Good afternoon. First of all, in the name of the President of IPV, I want to welcome all the participants to this workshop. During the next days, students, researchers, and professionals will have the opportunity to listen experts in the field of entomology and plant pathology. So I hope that all participants get the most of out the talks, both in the online webinar and in the in-person workshop. I also want to thank all the invited speakers and the organization of this workshop. Our research center, CIMU, and our associate laboratory, SUSTEC, develop research activities of great relevance in this area. So it is very important for IPV to join all these experts. Thank you very much, you all, and have a nice webinar and in-person workshop.
Thank you very much. Good afternoon. I would like to say hello to all the participants and welcome. For me, it is a great pleasure to welcome you for this workshop. The workshop will occur during 2 days remotely, and after that we will have another 2 days in person at the laboratories of the Polytechnic Institute of Bragança. I'm very grateful to Isabel and the other organizing committee, and also I'm very grateful for all the teachers from different countries of the world. And I would like to say thanks for the different institutions that support this workshop, namely the Polytechnic Institute of Bragança and their research center, the Mountain Research Center, the CTED program and their project, the IBER Chifres, and also the International Olive Oil Council and the European Food and Safety Authority. Just a few words about the Mountain Research Center. This group, the entomological group and the plant protection group, is a very dynamic group, and they organize different courses, participated in different national and international projects, and are very active in this domain of research. I'm also very grateful for all the participants. We have 150 registered participants from 7 countries, and I wish you a productive workshop, and I hope to see you in person in Bragança in the next times. For my side, I only to say, and I desire that you have a nice workshop. Thank you very much. Francisco, it's your time.
Yeah, sorry, I have had a problem with the connection to internet, but now I think it's all correct. Okay, welcome everyone. My name is Francisco Beitia. I'm a researcher at the Valencian Institute of Agricultural Research, located in Valencia, Spain. And I would like to thank the organization of this workshop for giving me the opportunity to participate as moderator. Today, we are starting the first session of the webinar, identification, bioecology, and management of insect vectors of Xylella fastidiosa. A two-day event that will provide an up-to-date overview of the plant pathogenic bacterium, Xylella fastidiosa, and its vectors. This workshop also includes practical sessions focused on insect vectors and potential vectors. The workshop is supported by CITED Ibercifas, Red Iberoamericana para la Vigilancia de Xylella fastidiosa, and by the International Olive Council. The participants in the webinar can post their questions in the YouTube chat during the talks, and at the end of each of one of these talks, there will be a debate between the audience and the presenters of the talks. Okay, so to begin the session, I would like to introduce the first speaker of the webinar, who is Blanca Landa. She is a researcher at the Institute for Sustainable Agriculture in the Spanish National Research Council in Córdoba, Spain. She has more than 25 years of experience in plant pathology and molecular microbiology. Currently, she is the PEXIL project coordinator, a project that supports studies in basic and applied research on the drivers of Xylella fastidiosa establishment in Europe, resistant plant varieties, new detection and surveillance systems, plant microbiome treatments, and economic and social impact of outbreaks of the bacterium. Blanca has several publications related to Xylella fastidiosa, and today she will tell us about the Xylella fastidiosa in the world and the situation in Spain. So please, Blanca, when you want, you can start your talk.
Thank you, Paco. Can you see now in the full screen?
Yeah.
Okay. So good afternoon to everyone. My role today is to give you a general overview or an introduction to the pathogen Xylella fastidiosa and what is the situation in the world, and more specifically in Europe and in Spain. So this is the outline of my talk. I will provide some general characteristics of the bacterium, biology and ecology. What is the host range of the bacterium already known? What is the European regulatory framework? A few ideas on the diagnostic tools, because you will see more in details in one of the talks in this workshop. And just focusing on the geographical distribution of the different subspecies and sequence type in the world, and the main crop affected by this bacterium at the European level and worldwide, and then just focusing on the situation in Europe. So as you know, the bacterium, the name comes from the place where the bacterium is located in the plant, the xylem vessels. The fastidiosa names to the difficulty of growing this bacteria in vitro in the laboratory. So it's a bacteria that has very specific nutritive requirements for growing in the lab. It has a broad host range of more than 600 species, but it is important to notice that only in a few, in a small number, cause severe disease symptoms and devastating diseases. It has a dual lifestyle. It is confined in the plant within the xylem vessel, so we cannot find the bacterium as epiphytic. And the other part of the life cycle is it happens within the vectors that transmit the bacterium. And this workshop is dedicated to them, so I'm not going to talk about this. Just to give you an overview, this bacterium started to be studied in the late 19th century by the Pierce, that he observed that there was some problem in the vineyards in California, and they were destroyed by a mysterious problem. He was sure that was a biotic agent, but he could not isolate or determine the real causal agent. This problem continues during that century and the following one. It was thought at the beginning that was a virus because it was transmitted by vectors. by science, but we have to wait till the late in the century, in the 20th century, when the Koch postulated could be demonstrated, the bacterium could be isolated. Finally, it was named Xylella fastidiosa. Nowadays, Xylella fastidiosa is still a problem in California, even it has been the focus of research for more than 1 century. Once in a while, we continue producing devastating epidemics in the area. See, here you have some of the old publications. The first one was published in Science, in which it was demonstrated by the Koch postulates that the bacterium was the causal agent of Pierce disease. This bacterium is positioned within the Xanthomonaceae family. Although it was considered non-motil because it has no flagella, and nowadays it has been shown that it can be moved because it has some pili that has the ability to allow the bacteria to move against even the silent flow. By the type 4 pili, and the type 1 and type 2 pili can help the bacteria to produce biofilm and aggregate in different matrices. The bacteria grow really slowly, so for those that we work at the laboratory with it, you need at least 2-3 weeks to get enough amount of inoculum of the bacteria, and it's very small. The colonies normally are very small. The optimum growth is between 6.5 close to 7. And it has different type of colonies. One are smooth and other are, as you see here, with more wrinkle appearance. Another important feature of this bacterium is that it can be divided in subspecies. The 3 ones that has been already identified officially are subspecies fastidiosa, subspecies multiplex, and subspecies pauca. Each has a different potential origin. Normally they affect different host plants, or at least they are more virulent in some specific host plants. Also, in the literature, we can find other subspecies as sandy, tusky, and morus, but as I told you, they have not been described officially following the international nomenclature of microbiology. So what is important to know exactly what we have in our territory, because we know that there is a risk of appearance of new variants of the bacteria. The bacteria can recombine and produce new variants that can overcome some resistance or cause disease in other plants that are already before they didn't origin a problem. So it's a mesophilic bacterium. The optimum growth is between 25-28 degrees. It's shown that it's sensitive to low temperature below 15 degrees. And also that the temperatures higher than 34 or lower than 10 degrees limit its survival in the plant. In our laboratory, Miguel Roman is performing a really nice work in which he is studying a huge collection of bacteria from all over the world belonging to the different subspecies. And what we have found is that the global range of optimum temperature is between 20-28 degrees. It's also there is a difference among the different subspecies and particular strains. So we cannot generalize the optimum temperatures for each subspecies or for Xylella fastidiosa as a global bacterium. The geographical distribution of Xylella fastidiosa is shown in this slide in different colors, the different climates. And in green dots, you have the places in the world where the bacterium has been described. So normally it prevails in areas with climate tropical temperate, but we can also find the bacterium in continental and semi-arid climates. As I told you before, the bacterium is only circumscribed to inhabit the silent vessel of the plant. And then it is needed that an insect, or if we do science, we take a portion of the plant and including in a new plant. It's the only way that the bacterium can be moved from one plant to another, or just by man with the movement of plants for trade. So when the insect feeds on the plant, in the infected plant, acquire the bacterium, it immediately after is able to transmit it. So if it fits in a diseased plant and then go to a healthy plant, it can transmit the bacterium. Within the plant, once the bacteria is inoculated by the insect, it can move upwards in the plant with the subflow, but also it can move to the roots of the plant and the below ground parts of the plants, but using the twitching movement with the PI4 type. And also the bacteria can move horizontally by using polyglucuronase, that are enzymes that can degrade the pit membrane that is present between the xylem vessels. Concerning the host range, in 2013, that was the year where the bacterium was declared in Italy, EFSA made a statement in which at that time it was known that 132 host plant species, 132 plant species were hosts of the bacterium. Soon after, EFSA continued doing and updating in different panels the host range, and as you can see here, in the different years, there was a really high increase in the number of host plants of the bacterium. And nowadays, every year or even twice per year, EFSA continue updating this database. These are some of the numbers by the different subspecies, in which you can see the big difference between 215, in which 300 host species. We have 300 host species. The last update that happens a few months ago, we have increased up to 664. One important point is that EFSA categorized the host plants according to the literature, depending on the detection methods that have been used by the researchers or reported in the scientific literature. So the most restrictive one, and the one that we can count more on, are the section AA, in which at least the bacterium has been determined by two methods. One of them can be symptoms of the test plant if it has been inoculated, by ELISA, by PCR methods, sequencing, and culture isolation. But at least one of all those methods has to be molecular approach. If we go to BCD, we are less restrictive for the detection of the bacterium. So the highest number, over 664, is regardless of the detection method. It is considered even a declaration of symptoms, but not a demonstration by molecular approaches. So if we want to be sure that the bacterium has a host in a plant, we should consider the A section. So these are the big difference in numbers. If we are very restrictive, we have 423 plant species in close to 200 genera and 68 families. And if we are less restrictive, we have over 679 plant species in 88 families. I have to tell you that this huge increase in the number of host plants also has occurred because the detection in Europe, especially in Italy, France, Spain, and Portugal, because after monitoring the territory in the demarcated area, we have been able to discover new host plants of the bacterium. And as I told you, this is not always developed in all hosts. So due to the monitoring, many asymptomatic plants are also sampled and analyzed, and we have determined that they have been infected naturally under natural conditions by the bacterium, but no symptoms are present in some of the hosts. So also an important point is that we have differences among the different subspecies and sequence type in the different areas. So we don't know yet if there is some strain host specificity in those areas, if the vector population in those countries or in those areas are different, or if there is some feeding preference by the vector population. For those of you that want also to be updated with the official host plan list, you can go to the European Commission portal, and there you can find periodical updates of this host range list. Moving to the European regulatory framework, as you know, Xylella fastidiosa was a regulated quarantine pest since 2000. And it was included in the list A1 of EPO, but since it was detected in Italy and confirmed, distributed and established in the region of Puglia, it was moved to the list A2 of EPO. Officially in the world, it has been described in 16 countries, although we have some evidence that it may be present in other countries, but they have not been declared officially. In Brazil, it has been declared in 11 states, 3 in Canada, 26 in the USA. In Argentina, it's also present at least in 2 regions. If we look into the climatic suitability models developed by ESA, in particular by my colleague Juan Antonio Navas Cortez, this study showed the higher suitability for the southern European regions of the European Union, especially Portugal, Spain, France, Italy, Greece, and Cyprus. So after the first outbreak, the official mandatory revealed that the bacterium was present in different areas in Europe, and as you can see, with a few exceptions, such as this in Germany, all the outbreaks were declared in areas that were suitable for the bacteria establishment. So since detection in Italy and due to the concern of the extension of the epidemics, the detection in other areas in Europe, EFSA, in collaboration with the Joint Research Center, developed integrated economic and pathological study, combining the expertise of both organizations. They make a study of including different indexes of economic, social, and environmental impact. Xylella was ranked first in 13 of these 25 indicators that you can see here, and it was considered the first priority pest for Europe among 20 pathogens and pests of importance for Europe. That was due because the economic cost of this bacteria, if it's completely full spread, will be very, very high. It was estimated about 5.5 billion euros per year. The bacteria can cause very important losses, especially in olive trees production, but also in the export losses of the union, not only the direct producer, the olive farmers or citrus, almond, and grape farmers, but also the export producers and the nurseries. If we compare the cost of production versus the cost that the union is using for surveillance, you can see that it's worth the effort that the European Union is doing every year. We have here, for instance, in 2019, less than 5 million per euros if we compare to the 5.5 billion of euros per year. Also, the social cost is very important. If Xylella could have a full spread, more than 300,000 jobs involved in the production could be at risk. Not only, as I told you, primary production, but also the nursery sector. Also, 70 agricultural products covered by European quality levels could be susceptible of this problem, and different plant species present in UNESCO World Heritage Sites could be under risk. And we cannot forget also the environmental cost of Xylella fastidiosa if it could spread across Europe, because We should increase the use of spraying insecticides for controlling the vector to stop the spread of the bacterium, and also that the natural habitats, protected species of birds, plants could be also affected. So this is the current situation in Europe. We have three countries with areas under containment: Apulia in Italy, Corsica in France, and the Balearic Islands in Spain. Then we have areas under eradication in the north of Puglia, in the Toscana region, in the Paca region in France, and in Occitania. We have Alicante, the Valencia community, and four areas, sorry, one is missing in Portugal. I have to say that the plant detected infected in Almeria and Madrid has been already declared as eradicated, because the legal time framework that it needed for that, it has passed. We have been able to show that after 2 years in Almeria and 4 years in Madrid, there is no more positive plants. So what happened when we declare an infected plant? So is infected zone is declared in the 50 meters radius, and all the plants declared as a specified host in that area has to be removed, irrespective of it has the show symptoms or not. Then we have a buffer zone that it depends of the situation. If it's an isolated case, that means a new case, we have a buffer zone of 1 kilometer. If we are in an area under eradication, we have a 2.5 kilometers radius, and the area under containment, we have a 5 kilometers radius. So what is the Union doing for the diagnostic tools? So in this map, we have the distribution of Xylella fastidiosa in the world. And you see here different points, different dots with different colors. Each of them refer to a different subspecies. So it is important to know that in Europe, we have a genetic diversity. We have different subspecies of the bacterium. And as I told you, it is important to know the subspecies because each of them may have a different host range. Knowing the subspecies and the genetic relationship, we can determine what is the potential origin of the introductions. For instance, Pauca, we know that comes from Central America, from Costa Rica, and then we have multiplex and fastidiosa in Spain and in France, that the potential origin is from California, etc. The legislation from the European Union, especially the Commission implementing regulations, declare what are the tests that are official for the identification of the bacterium. So we have in Annex 4 identified what are those tests. So we have one only to determine if we have a plant infected or not by Xylella. These are the tests for screening. And we have the real-time PCR by Harper and Ojan. We have the LAMP assay by Harper and the conventional PCR by Minsavage. And then we have other set of molecular tests that are used for identifying the subspecies that we have present. And the most popular one, and the one that is used by Europe, is the multilocus sequence type. In Spain, this is the normal procedure. So the regional laboratories are the ones that need to do the surveys and test the plants. So we have the official plant health laboratory in Spain in all the communities, in this case, Balearic Islands, Comunidad Valenciana, because we know that the bacteria is present there. So when they find a positive plant, this sample is sent to the National Reference Laboratory in Valencia, in IBIA, led by Dr. Marco Noales. It has to confirm this positive plant, and also provide support and training for the laboratories. Once this laboratory confirms the positive, a DNA sample is sent to our laboratory in Cordoba at the Institute for Sustainable Agriculture, and we do the MLST typing. And then we determine the subspecies and the sequence type that is present in the sample. All the protocols, if you are interested, can be found in the EPO protocol. I'm not going to explain you because you will see more in a different talk. But just to know you that the Xylella fastidiosa diagnosis originally cannot be determined by symptoms or isolation because are not affected at all due to the unspecific symptoms of the bacterium and the slow growth. For hundreds and thousands of samples, for instance, in Italy, it was used ELISA because the cost was much cheaper. But nowadays, that the PCR price availability of equipment in different laboratories, most of the plants are analyzed by PCR. So there are several, more than 20 PCR protocols available using conventional or real-time PCR. Very recently, two of them, based on tetraplex, that can identify 3 subspecies at the same time, have been developed. Probably these 2 methods will be included in the next EPO update protocols. Now we are also moving to the diagnosis of the bacteria fast, because if you go to the field, you want to know exactly if the plant is infected or not, or even used for insects. So the LAMP recombinase polymerase SS are also used. The digital PCR that is very sensitive and less prone to effects of inhibitions is also being implemented in different laboratories. And the last methods that are more updates are the use of whole genome sequencing, either using Illumina or nanopore technologies. In our laboratory, we have developed a system based on capture, hybridization capture. So the idea is to, from the whole genome of the plant, try to fish only the sequences that belong to the bacterium to make a more effective diagnosis of the plant based not in 7 genes as we do with MLST, but based on hundreds of genes. So, as I told you, this is the official method for subspecies determination in the European Union, that if it is based on MLST. So you have to amplify 7 genes, sequence them, and then obtain the 7 alleles to determine the sequence type. That currently, worldwide, there have been 90 different sequence types identified in all the countries. This approach has been also used to identify potential recombination events between subspecies. And in the case of subspecies morus, it has been shown that was originated by the recombination between subspecies multiplex and fastidiosa. So what is the geographical distribution of subspecies and sequence type? So subspecies fastidiosa, it is proposed that was originated in Central America, especially in the area of Costa Rica, and then was moved to the United States. So it has more than 20 sequence types described worldwide. Most of them are in Costa Rica, the biggest number, 4 in the USA and 1 in Mexico. There has been also the recombinations between fastidiosa and sandy. 2 of them has been described in Costa Rica and 1 in Mexico. Concerning subspecies multiplex is the one that has more sequence type. Nowadays there are more than 37, I think, close to 40 sequence types described in the world. Most of them in the USA, because it is proposed that this subspecies was originated in Northern American continent, 3 in Brazil and 1 in Argentina. Concerning pauca, that the origin was proposed to be South America. More than 20 sequence types has been described, most of them in Brazil, 17, 3 in Costa Rica, and 1 in Argentina. For sandy and fastidiosa sandy, you see here the events are less numerous. We have only a few in those areas. And for subspecies morus that was originated in the USA, we have a few, four sequence types described in the USA. The subspecies Tasky in the past was proposed to be present in this area in the USA, but no one has been able to isolate this subspecies anymore. There is no strain available, so there is some controversy of the real existence of this subspecies. So what is the situation in Europe? As I told you, many different introductions occur in Europe, and we have 4 subspecies identified and 11 sequence types. You see here the distribution in the different countries. So the highest diversity is in the Balearic Islands and in France. Then we have different sequence type in Spain, the one that had been described in inland Spain compared to the ones described in the Balearic Islands. And in Portugal, so far, only subspecies sequence type 7 has been identified in many different hosts and plants, and it seems that one sequence type from fastidiosa has been identified, but is not officially declared yet. So what is the importance of knowing the sequence type? Because as I told you, the mandatory management of the European Commission are based on the subspecies present in each outbreak. So that means that the monitoring and eradication depends on the subspecies and the sequence type. And the list of host plants affected in each regulatory framework are different depending on this genotype of the bacterium. And as I told you, the assignation is based mainly on MLST analysis, but this has a limitation, as you will see later. So I think that in the following years, the use of whole genome sequencing will provide a higher phylogenetic resolution and help to infer important biological and ecological features. We have working on laboratory also in developing in collaboration with the laboratory of Maria Agnès Jacques at INRA in France, a protocol focused on nested MLST analysis. So using this approach, based on forensic analysis of ring tricks, we were able to identify different rings infected by the bacterium in almond trees. And what was more important, we were able to identify that the bacterium, Xylella fastidiosa, on those trees were present in years that could go back to the 1998 or 1996 for fastidiosa and multiplex, showing that the infection of the bacterium in the Balearic Islands took place years before than we even thought. Also, using the whole genome sequencing of some isolate, we were able to show that the subspecies multiplex in Europe was originated from isolate from California. And that in Europe, 5 different introductions, at least 5 occur, one independent in Italy, in the Toscana region, ST87, 2 in Corsica, ST6 and 7, 1 in Balearic Islands for multiplex, ST81, and that this was different, the one in Alicante, Spain, ST6, that is here, is completely different from this one in France. So even the MLST analysis show that all isolates belong to the same sequence type 6. They are completely different strains. So the origin of those groups of bacteria are different. As you see here, there is a lack of monopoly for the isolate assigned to this sequence type. Using this hybridization capture protocol that we have developed in our laboratory, we have been able to precisely reconstruct the phylogeny of Xylella fastidiosa using blind trials in which researchers from different laboratories send us DNA from outbreaks or from inoculated plants artificially in the laboratory, we were able to identify, for instance, sequence type 2 from plants that were provided by France, sequence type 16 or sequence type 74 provided by researchers in Brazil. And what is more important, if you pay attention to this cluster that is from subspecies multiplex, we show that as I told you, isolates from Spain and France were completely located in different groups. And that sequence type 81 from Balearic Island was closer to sequence type 6 from France. And another important point was that the isolates from Portugal, France, Spain, and a few isolates from California, from ST7, are very similar. But we can find sequence type 7 from California here and from California here that are different. So it seems that the sequence type 7 present in Europe at least came from the origin from the same region. It doesn't mean that the introduction event happens only one. It can happen several times, but the genetic relationship of this genetic group is closer from all the isolates in Europe belonging to this sequence type. So moving to the main crops affected at the European level by Xylella fastidiosa, we need to take into account the disease triangle that all the plant pathologists and epidemiologists study. So in order to develop an epidemic as severe as in Italy, all the components of the pathosystem have to be far above all. So for instance, we need a strain of the bacterium that is highly virulent. For instance, Pauca ST53, that is highly virulent on a susceptible host plant, that is olive. And in the case of Puglia, the most cultivated or the highly grown cultivar, Xylella salentina, is very susceptible to this specific strain of the bacteria. Then, in order for the epidemic to spread so fast and in such big extension, we need a vector that is effective. That also it is a very high population levels. And then, of course, the environment has to be conducive for the disease to develop. So the most important diseases that this bacteria can cause are the first disease on grapes, the citrus variegated chlorosis in citrus, the dwarf in alfalfa peaches, and then we have different leaf scorch diseases that have been described in almond, oleander, olive, prunus, blueberry, and cherry. But also the bacteria can cause important problems in oak and different forestry trees. So these are typical symptoms of pear disease on red wine on the left and white wine varieties on the right. And typical symptoms that the good maturation is not even. You see here green parts with the more developed parts, and we have this defoliation of the leaves. It is interesting that in grapes, so far only isolates from subspecies fastidiosa has been described. In the USA, Spain, sequence type 1 in Spain, that is coincident with the one in the USA, and a few isolate sequence type in Costa Rica. But we don't have any isolate from other subspecies that have been obtained from grapes under natural conditions. In citrus, these are the typical symptoms on the leaves and a reduction of the size of the fruits. Some of the genotypes that cause problems in citrus has been also isolated from coffee, but normally they belong to different populations. So the sequence that can infect citrus are not similar to those that can cause the crespero coffee leaf scorch in coffee. So in citrus, these are the sequence that has been described so far. You have most of them are from Pauca. We have sequence type 1 and sequence type 2 from fastidiosa. So it is interesting that although this sequence that has been described in fetid citrus, normally they are not associated to big problems as a difference the ones that the symptoms that are induced by the subspecies Pauca. So we need to still to know what can be the real significance of finding sequence type 1 in citrus. For coffee, there are many sequences that have been detected infected in coffee plants. We have many from fastidiosa in Costa Rica, all of these, in Brazil. And also there are many reports of intercepted coffee plants in the frontiers. In Europe, coming from different countries such as Costa Rica, Ecuador, Honduras, and Mexico. And this intersection made to ban the imports of coffee plants to the European Union. Concerning olive trees, you have here some of the symptoms that the bacteria induce in Italy. It was shown that the bacterium was present in California, causing these symptoms in 2011. Also in different regions of Italy, and in the same years, it was also detected in Argentina and in Brazil. And the sequence that can infect olive are also very diverse. So it seems that the olive is a very suitable host plant for Xylella. So in the USA, ST7, ST81 from multiples were detected. In Italy, ST53. In Brazil and Argentina, in the first years of the study, we have only one of each. In Spain, we have identified ST6, 81, and 80 from multiples and from Pauca. It seems that in Mexico, fastidiosa was also isolated from olive trees, but the symptoms were not very clearly defined. And now in Brazil, since the X Factor project, a deeper study was made on olive plants, and they found that different sequences that from Brazil could also infect olive trees. In Portugal, there is one case of detection of ST7 in olive. And recently, this new sequence type ST90 that is quite unusual because it seems a recombination between fastidiosa and Pauca has been detected in Argentina. Concerning almond trees, these are the symptoms that you can observe in the field in Mallorca Island here. Here we have on the bottom in Alicante. These are severe symptoms occurring at the end of summer. They are called the golden death because the tree almost collapsed at the end of the season. So there is also a huge diversity of sequence type from different subspecies that has been described in almond trees in Brazil, France, Spain, the USA, and Italy, and recently also in Argentina. So what is the current situation in Europe? So as you know, the bacterium was declared officially associated to these three symptoms at the end of 2013 in this area of Gallipoli. These were the symptoms that the bacteria was originating at that time. Here you have the speed of the progression of the symptoms in the canopy of the tree. You have a Google Map picture in 2013 and the same place in 3 years earlier. So you see the big impact on the trees. Even in the same year, you have picture from February and the same place in November, you have complete collapse of the trees in a few months. And this is the picture that you can see in internet. If you search for Xylella fastidiosa Italy, 2013, the plants in 2016, 3 years later, in monumental millennial trees in 2 years, you see the big impact. And here you see what is the situation in this area 2 years ago. This epidemic that was not seen anywhere, at least for this bacterium, focused attention of very important journals such as Nature and Science, and many articles and interviews appear on those journals. And here you have, if you go by car in the area, and this is not a very highly affected place, but you see here how it is spread all over the orcas. So all the canopy is affected in more or less extent. And even some trees have been severely pruned to try to rebuild the canopy of the tree. Sorry. So currently the demarcated area in Puglia overpass this region. So it is estimated that more than 700,000 hectares within 140 kilometers are affected. And the number of olive and the rigs is said 25 million of trees that comprise more than 50% of the territory of Puglia. The thing is that the demarcated area, the area that was it was fixed free of the bacterium, new foci has been found in the north. So new trees continue appearing northward. More than 60 million of olive are thought to be potentially affected. So the new demarcated area is moving, as you see here, according to the new outbreaks that has been detected close to Conversano and Mola di Bari, that are the small municipalities. And here you have this real data in the webpage of Emergenza Xylella. So if you go into the webpage, you see all this green area and this red point that are the positive. So if you dip into the scale, all these green dots are plants that have been monitored for the presence of the bacterium. So you can have an estimation of the thousands and thousands of plants that are being evaluated every year. And here are the ones that have been detected infected. So these are the last two outbreaks detected close to Poligliano Amare and close to Monopoly. This is really sad because at the first time that the bacterium was detected, you need to drive from Bari to the outbreak area for 2.5 hours. And now, if you leave Bari, you can reach Monopoly in less than 15 minutes. And of course, in the newsletters, newspapers in the area, you can see every week different articles reporting the huge number of olive affected, the economic losses, and that a small olive mill are closing. Concerning the host plant in Italy, so far 36 host plant has been detected infected by the bacterium, and all of them only infected by sequence type 53. So there is a huge homogeneity of the population of the bacterium. And most of the plants are natural vegetation, a few ornamentals, but only 4 plants could be considered of economic importance: Olea europea, Prunus avium, Prunus dulcis, and Pistacia vera. Interestingly, although almond and cherry has been infected by the bacterium, so far no citrus plant or grapes had been found infected by the bacterium. In Italy, there was also another outbreak detected in the region, in the Toscana region, in this peninsula. It was at the end of October. This is under eradication. Up to 24 host plants has been found and infected by the bacterium. Only almond and fig as cultivated crops have been found infected there, and the rest are plants from the natural vegetation typical from the Mediterranean climate. In this area, only this sequence type 87 that was new for the science were detected. The situation in France is that more than 70 species, mostly of ornamental plants, are host for the bacterium in this country. In Corsica, we have the containment strategy, and here we have detected mainly sequence type 6 and 7, and only one foci has been found for ST53 or ST76. In Paca region, that is under eradication, we have found the same sequence types 6 and 7, and also a few cases of new sequence type 88 and 89 recently described for science. And in one case that was really concerned because was ST53, the same from Italy, and it was found in a garden in olive trees, but was eradicated. In Occitania, we will see now in more detail. So far, only sequence type 6 has been detected, and the strategy is eradication. In this region, we have found only one vector, Philenus espumarius. I forgot to say that in Italy, 3 species of vectors have been identified, Philenus espumarius, Neophilenus campestri, and Neophilenus italicus, if I'm not wrong. So these are some of the host plants that have been found infected in France. Here you have the list. So most of the plants belong to the Mediterranean maquis, so it has been established in the natural environment, especially in Corsica, and only a few has a regional economic impact, such lavandula and curry plant. Here you have some of the symptoms of olive trees that, although officially only one case of an olive tree has been reported in Corsica, there is many cases of this typical decline of olive trees that is not clear if it's associated or not with Xylella. And here you have more cases of controversial olive trees in the island. So a recent genetic analysis performed by the group of Maria-Agnès Jas at INRA evaluated the genetic diversity of the isolate from France, and they found that they belong to sequence type 7, that were closer to sequence type 6 from Alicante, and the sequence type 6 from France that were closer to the ones from Balearic Islands. And here you have in course in blue and Paca in red, sequence type 6 on the top and sequence type 7 on the bottom. So it is thought that 2 founder populations, one from ST6 and one for 87, spread and cause some small diversification of these 2 populations between Paca and Corsica regions. Concerning the vector, a study that was conducted in Corsica found that Philenus espumarius, as I told you, is the only vector found infected by the bacterium. Most of the insect specimens were contaminated a similar proportion by both sequence types. So there is no preference for any of them. An important point was that within the insects, some alleles were found that belonged or could be assigned to Xylella fastidiosa, subspecies fastidiosa. So although the bacterium has not been identified in plants, it is some kind of interesting that the vectors had fed in plants that harbor alleles belonging to subspecies fastidiosa, but they don't know yet what can be this origin. And this is the situation in Occitania. It is an area close to the frontier with Spain. So people in Catalonia are really concerned of this situation because as they start with the monitoring of the region, they are finding more outbreaks. And for instance, in the Out and Arriere department, they had found more than 1,000 plants already infected by the bacterium. And here you have some of the plants that have shown positive. We have Medicago, Spartium, Salvia, Titisus, a few cases of Prunus dulcis, but also Laurus nobilis, Platanus, and Lavandula. And this list of host plants are new for science, so it has been only described so far here in France. In Portugal, I'm not going to talk because you will have the possibility of listening directly from the Ministry of Agriculture in your country. But there is also big concern because there have been 4 main forces identified in the country. And the most important thing is that more than 78 host plant species has been detected infected by the bacterium. And some of them include very important plants such as citrus species, lemon, mandarin, tangerine, orange, beeties, and different quercus, very important for forestry and for natural areas in Portugal and in Spain, and 2 prunus species. So just finishing with the current situation in Europe, I will move to the case of Spain. So as I told you before, we have 2 different situations in Spain. The Balearic Islands are under containment since December 2017, and Alicante is an area under eradication. So we have different genetic diversity in the islands. What is present in the Balearic Islands is different of what is present in Alicante region. So the first plant detected was found in a garden center near Manacor. It was a cherry plant. And then when the survey started, more positive were found close to this region and in the different areas. So this is the number of positive and host plants that has been found as the monitoring has been progressing. So that not mean that the bacterium has spread during this year. Is that when the exploration of the territory took place, we had found different positive plants. So the current number is close to 40 because there are not official declaration finished yet. But the last data that was from January was 38 plants infected by the bacterium and more than 1,000 positive plants. So these are the positive plants. Most of them are from grapes, olives, and almonds. In the islands, we had found Philenus spumarius and Neophilenus campestris infected by the bacterium. And we have, in Mallorca, we have subspecies multiples, sequence type 7 and 81, and subspecies fastidiosa, sequence type 1. In Menorca, only subspecies multiplex sequence type 81. And in Ibiza, we have a different situation that we have subspecies pauca sequence type 80. And this sequence type is the single place in the world so far that we had, it has been described. And these are some of the host plants that have been found infected, grapes, almond, cherry, plums, olive, wild uncultivated. We have figs, juglans. We have ornamental plants such as polygala, lavandula, rosmarinus, fraxinus, acacia, nerium, cistus, rhamnus, genista, teucrium, calicotome, cistus, etc. So many plants from the natural environment typical for Mediterranean vegetation. So, as I told you, we are the national reference laboratory for identification of the different subspecies. So, in collaboration with the government of the Balearic Islands, we have a project to study in detail the genetic diversity present in the island. So, the procedure we follow was to perform the MLST, the conventional procedure, declared official by the European Union. So, if you amplify this gene, you follow with the conventional procedure. But if you are not lucky and you are not successful in the amplification with the conventional procedure, we use this new procedure based on nested MLST. And then with this procedure, we are able maybe to amplify the 7 genes and to assign the isolate to the sequence type. Or if we are not lucky enough, you can only identify the subspecies or nothing. So here you have the percentage of successful amplifications in blue for the different genes in simple PCR or nested PCR. So you see that some of the genes, such as cis, are the most successful, whereas the MALF-F is one of the less successful. Nevertheless, if we use this combined approach, simple and nested MLST, we were able almost to amplify 100% of all the genes in all the samples. And here we have for 400 samples analyzed in 2.5 years. We were able to identify the subspecies almost in 100 samples, more than 95 assigned to sequence type. And what we found is the presence of the different subspecies in the Balearic Islands, confirming the results that were previously reported by different laboratories. So these are the distribution of the different host plants in the different islands, Mallorca, Menorca, and Ibiza, and the different subspecies sequence type. So all the cases that are in yellow are new cases reported thanks to this project that was financed by the tourism in the Balearic Islands. So maybe you go to different countries and you pay for a tax that is called touristic tax. So in Balearic Islands, some of the money that came from this tax was devoted to science and to do research on Xylella fastidiosa. So all these analysis and other projects were financed by by the government of the Balearic Islands. And here there is a continuation also of the different hosts and the different sequence type. So mainly, not always in the different islands, you can find the same subspecies or the same host plant infected by the different subspecies. And again, you have here these symptoms that you saw already. In olives, I have to say that the multiplex induce less severe symptoms as compared to those caused by Pauca in Ibiza and Pauca in Italy. And also you see here the desiccation of white olives that are very affected in Mallorca and Menorca. Also you have some pictures in cultivated or white olives. On grapes, on red varieties and white varieties, desiccation of the resin of the fruits. These are some figures of the number of plants eliminated, eradicated in the Balearic Islands. More than 30,000 plants have been eliminated. You have to remember that the island is not under eradication anymore, so it's under containment. So most of the plants, this huge number came from the first years that were under eradication. Of course, the movement of plants, there is restriction. You can find these levels on the airports. The islands cannot export plants to any place, host plants outside the islands, but also within the islands, there is some restriction of movement, especially from Ibiza to any place, to avoid that Pauca that is considered more virulent can move to Mallorca or Menorca. And the situation in Alicante, the first outbreak was declared in summer of 2017, and soon after another outbreaks were detected close by, a few kilometers distance. So these are the two first outbreaks. As you can see here, the plot, the orchard was eradicated really soon. You see here a picture before and after the eradication. In this area, only sequence type 6 has been detected. And the current demarcated area is over 135,000 hectares here. All the positive are marked in yellow. So it affects 76 municipalities in the demarcated area. 2 of them has already been declared in Valencia province, so it's not only Alicante, but 2 in Valencia. We have more than 1,000 positive. So indeed, total plants already were all data close to 5,000 positive. In this area, 3 vectors have been detected positive: Neophylenus campestris, Neophylenus lineatus, and Philenus spumarius. Most of the plants found infected are almond plants, more than 90% of the positive. And here you have some of the positive plants. Again, we have some Prunus species, Prunus dulcis, Armeniaca, and Domestica, but many plants belonging to the natural vegetation of the Mediterranean landscape. Here you have some data provided by the Generalitat Valenciana. These are the positive detected since 2017 to 2021. So as you see, about 90% of the positive were almond, followed by Cotome, Helichrysum, and Polygala. And some data of the positive, the vectors that have been shown positive. The number is really low, lower than 5% of insects declared positive. Here you see how the monitoring occurs. So the area is declared in different sampling areas. In red, the ones that have been monitored and with positive. In green, those that has been already tested. In gray, those that are already pending. So what they are doing is the priority for eradication starts, this is the border, the free area. So the positive that first are destroyed are outside the demarcated area, close to the free bacterium area. Then in the area in orange, and latest the area in yellow. Of course, all the Valencian community is being monitored accordingly. So more than 100,000 plants had been analyzed in the last 4 years. So when a plant is found positive, so some insecticide treatment are applied before eradication, and the plants are destroyed. The tissue is ground left on the ground. Of course, there is a lot of movement against this procedure among farmers, and there are requirements following the law to go against these circumstances. And also even there is some sabotage of the equipment that is used for eradication. So the situation is similar in some way to those shown in Italy in the first year in the Salento region. And also there is the demarcated area, there is the movement of plants banned, and you can find these labels all over. What is interesting if we compare the host plant that infected by subspecies multiplex in Alicante and in Mallorca and Menorca, the 2 places in Balearic Islands. Certain hosts are shared among the different regions, but in Alicante, there are also unique species found infected in the area. In Mallorca, Menorca, also 14 species are infected by subspecies multiplex. Very important point is that only in Alicante, although in Mallorca and Menorca, olive tree has been found infected by this sequence type, it doesn't happen in Alicante. So there is no need to eradicate olive trees in the area. Just the case in Madrid, I won't tell you, but it was an olive tree declared positive, only a single tree in a plot. We never know where it came from because the traceability of the plants was not possible due to the, it was a few plants bought in a nursery. After 4 years, there had been no more declaration of positive plants. So I think officially it will be declared free of Xylella really soon. And in Almeria, also there was a case of different polygala plants found positive. The situation here was better because the plants were grown under a screen proof for insect. So only 2 years were needed to declare the area free of Xylella. And this is the official monitoring that the regional government is performing in the area. Of course, there is a big effort in olive plants, followed by citrus plants, prunus, and grapes. And just to finish, I want to tell you that after detection of the bacterium in Italy and in Spain, there had been a big effort in research in our country. So we just finished a national project that was financed by the INEA and the Spanish Research Agency, and also we received funding from the Interprofesional del Aceite de Oliva Español, so the olive farmers, the organization supporting them, also support our project. There were 13 research centers involved, 3 research centers from abroad, and more than 40 researchers, and the budget was over 1 million euros. In CSIC, that is the organism I work with, the Spanish National Research Council, we have a thematic interdisciplinary platform focusing on Xylella fastidiosa, in which we want to bring together all the knowledge, the scientific and the technical knowledge to search for innovative solutions to control the bacterium or the vector. Here you have what we are approaching this. We are focusing in the different components of the pathosystem, the bacterium, searching for antimicrobials, the host plant, searching for the exploitation of the plant microbiome, the natural resistance of the plants, also in the vector, to search for more sustainable ways to control the vector, and also in epidemiological aspect, searching for how the environment can affect the epidemics development. And just to finish, the BC project is a European project recently funded. We started the project in September. Our idea was to support the research needs. Even we have project, the European project that focus on Xylella. There were still many questions to answer provided by the farmers, the nurserymen, the managers, and the legislators. So based on those research needs, we developed the specific objective of our project that was divided in 9 working packages, in which we want to provide answer to a general impact and outcome of the project. So we want to enhance the capacities to prevent, monitor, and find adequate responses to this bacterium, to enhance the capacity to control the Xylella, mainly by biological approaches, focusing on the host plant, on the vector, on the bacterium, as I told you, the different components of the pathosystem. And also a very important impact that we want to provide is the provision of scientifically based support for relevant European and associated countries to support the plant health policies. As a difference, in previous European projects were focused on olive, but we know now that Xylella is not only a problem in olive, but also in other crops such as almonds, natural vegetation, the landscape of the Mediterranean, and the grapes. So BCIR will focus on other crops. Of course, we'll continue the research on olive. And this is the consortium. We are 31 partners, 28 beneficiaries will receive money from the European Commission from 12 countries, and 3 are associated partners from UK and Australia. It's a 4-year project, over 7 million euros. We include countries where Xylella fastidiosa is present in Europe and in Americas, and also Israel is included, but also countries where Xylella fastidiosa is not present. And from Spain, we have a huge consortium. So as Xylella is a very important pathogen for our country, we have been able to include different institutions from CSIC and also from different organizations within research centers and universities in Spain, and also the collaboration of private companies. And just to finish, I want to thank my research group and the team in Córdoba that support our research or different projects, the 4 PhD students that are conducting their thesis in Xylella fastidiosa related areas, all the financial support that we have received from different organizations, national funding, European funding, also even international funding, many different researchers that provide support to our research. And now we'll be happy to answer to your questions, so we can start if you want. And thank you for your attention.
Okay. Okay, Blanca, thank you very much for the presentation. I think it's full of information and very interesting. And possibly for this amount of information, possibly at the moment there are no questions, because possibly the audience is analyzing all this information. But I have at least 2 questions to you. The first one is a fundamental aspect in dealing with Xylella fastidiosa possibly is this relation between the different subspecies and STs and the host plants. Because, for example, in the case of the Valencian community, we have found that in Alicante, in the Marcached area, the fastidiosa multiplex, the subspecies multiplex and the ST6 is present on almond, but only on almond, not in olive trees that possibly are near these almonds. But in Madrid, although it was only a very particular case, but the same subspecies and ST was on olive trees. What do you think about this specificity between subspecies and host plants? Why? Is it real or not?
Yeah. The thing is that defining a sequence type is based only on 7 genes. So if you compare them, the whole genome, as I show in some of the phylogenetic trees, the sequence type 6 from Alicante is different from the sequence type 6 from France. Okay, so it can happen that the single case that was detected in olive, although was sequence type 6, was something different from what is present in Alicante. The problem was that we don't have enough DNA and we cannot recover the isolate to make a deeper genomic analysis. So if you focus on 7 genes, something can look like similar, identical, but if you broad and open the number of genes, you will see that there is much difference. So my opinion is that we have to move from the concept of subspecies, sequence type, to something more unique that is present in an area. Because even within the same sequence type, we can have differences for host range, etc. I know that that was a lot of controversy with the auditors from the European Commission because they could not understand at the beginning if it's subspecies multiplex and in Balearic Island infect olive trees and you have subspecies multiplex in Alicante, you have to eradicate also olive trees. So it took a lot of effort to inoculate olive plants and to show that really the strains from Alicante were not really aggressive on olive and were almost not multiplying at all. When you inoculate them, almost there is no multiplication. So, and this was, this forced the change of legislation because now what is indicated in the decision is that only the host plants that has been found infected in an area are the plants that should be eradicated. So until we don't find any olive with symptoms or with really infected by the bacterium, it's not considered a host plant. Okay, because of course, artificially, if you inoculate the plant, you force the bacterium inside the plant. Maybe the bacterium infect the plant, but under natural conditions, even the vector is not good, or it cannot transmit the bacterium because it's not a host for the vector and for the bacterium. Yeah.
And another thing is, I don't know if you have detected this, but at least personally, I have detected in the two last years that possibly is a decrease in the social interest in Xylella, in the problem of Xylella. I don't know if it's the case of the Malaysian community because it seems that the marketed zone is not it's the same for the last year. I remember that 3 years ago, there's more people, many people talking about the situation, many interested with talks, but I don't know. It's my personal impression that today the social perception of the problem with Xylella is not so high that some years before, and possibly this is a problem because the bacterium is present in the field. I don't know if you think something like that.
I mean, the social media has a very important role on this. If you go ask to the farmers affected by the problem in Alicante, they will say it's exactly the same. But this is not a fashion, but for instance, when the citrus plant were found infected in Portugal, there was another boom. The social media, the newspapers, the journalists started to call me, to call you, many people, because it's like a peak, you know, if there is a new detection, a new event, something new, so there is an explosion on the media concerning this problem, but the problem is still there. I mean, what is true is that at least in the last years, even the effort of analyzing the demarcated area, the buffer zone, only a few positive cases has been found. So in my opinion, it is now we have a more clear picture of the real situation in Alicante, and we know exactly what is the affected area. So now the effort should be placed on trying to stop the movement from this area outside. So I think the strategy that the government is following, starting the eradication from the outside to the inside, is the most intelligent one. But the problem is still there. People will start to live with the idea, to live with the problem, to start thinking in other ways of reconversion of the agriculture, or finding new alternatives, or even the government will provide subsidies for new plantations. I don't know. So, but the problem is still there. Yeah.
Okay. Well, from the organization tell me that there are no questions. So, well, many thanks for the presentation. I think it has been very, very interesting and full of information. A good presentation of the situation around the world. So, has been very, very impressive. Thank you, Blanca.
Thank you, Paco.
We will continue with the with a webinar. Okay, so our second speaker of the day is Paula Baptista. Paula is currently an auxiliary professor at the Polytechnic Institute of Bragança, Portugal, and a member of the Centro de Investigação de Montaña. She is the General Secretary of the International Organization for Biological Control, and a member of the Plant Health Panel of the EFSA, the European Food Safety Authority. Her main interests are in the field of applied microbiology, biotechnology, and agriculture, focused on the bioprospection of biological control agents and on biotic interaction. And she also has several publications related to Xylella fastidiosa and its vectors. And today she's going to tell us about the microbiome of Xylella fastidiosa vectors. So when you want, Paula, you can.
Okay, thank you very much for the introduction, and good afternoon to all. First, I would like to thank the invitation to be here present and to share with you recent results that we get in our group within the CIMO Research Center related with the microbiome of insect vectors of Xylella fastidiosa, in particularly Phylloenus spumarius. So I will share my screen. I hope that can you see it, please?
Yes.
Yes.
Okay.
Okay, so as I mentioned before, so I will present some recent results regarding the microbiome of Xylella fastidiosa in insects, in vectors that of this pathogen. Okay. So, as you know already, so Xylella fastidiosa is a bacteria that colonize mostly the xylem of the plants, and this bacteria is transmitted exclusively by vectors that feed on sap xylem. So, the process of infection starts with the feeding of the vectors of infected trees. So when they feed from infected trees, they acquire the bacteria that remains restricted to the foregut parts of the insects. That will so the one particularity of this bacteria is that this bacteria doesn't have the ability to colonize the whole body of the insect, so they are restricted to the foregut parts and colonize these parts and persist only in these parts. And once acquired, the bacteria can be transmitted immediately if these insects feed in a healthy plant. So in this process, the bacteria does not require a latent period before transmission. So after acquisition, immediately after acquisition, it can be transmitted for healthy plants. Both nymphs and adults have the ability to transmit it. However, the nymphs, they lose the ability they keep the infection capacity. It's lost during the molting process, so they can acquire the nymphs, they can acquire the bacteria, but then after the molting they lose the infection. And in what concerns the insect, once infected, the insects will keep the bacteria and can transmit it by feeding in healthy plants. However, the adults, they don't have the ability to transmit it, the bacteria to the progeny. Okay, so it must be also infected once adult. Okay. There is no apparent specificity between a particular Xylella fastidiosa subspecies and an insected vector species. So potentially all the insects that feed from Xylella, they can be infected by Xylella and transmitted. In what concerns of insect vectors, so potentially all the Xylella sap feeders insects can transmit the bacteria. However, the most reported insect vectors of Xylella belongs to the suborder Auxenoricha and to the infraorder Cicadormorpha, in particular to the superfamilies Cicadoidea and Cercopodoidea. In particularly the families Cicadoidea and Afroforodea. In Europe, we know that it has been reported more than 19 species as a Xylella sap-feeding insects. However, so far, only 3 species belonging to the family Afroforidea have been described as vectors of Xylella fastidiosa. In particularly, is Philaenos spumarius, Philaenos italosignums, and Neophilaenos campestris. So they are so far the most important Xylella sap-feeding insects that are vectors of Xylella in Europe. Among these 3 species, Philaenos spumarius was reported to be the most, one of the main vectors of Xylella in Europe. The insects, as all other organisms, we know that all the insects are colonized by microorganisms. Okay, so these microorganisms can establish association with these insects, which impact could be beneficial or have a detrimental effect on the host. Most of these microorganisms, they are symbionts, okay? So it means that they establish a symbiotic association with the insects, and both partners have advantages in this association. The symbiotic microorganisms in the insects, they play essential roles for the insects. For instance, some of these symbiotic microorganisms can manipulate the reproduction of the insects by promoting the feminization or cytoplasmic compatibility. But there are also other microorganisms that play an important role by supplying essential nutrients, and this is really very important in the case of sap-feeding insects, because as you know, the Xylella is very poor in terms of nutrients, and these insects, they rely their survival in getting essential nutrients provided by these microorganisms. So these microorganisms are really very important for the survival and the fitness, in particular, of insect feeders, from insects that feed from Xylella. Besides nutritional and reproduction services provided by these microorganisms, there are also other microorganisms that play an important role by protecting the insects from biotic stresses and abiotic stresses, namely temperature. And in terms of biotic stresses, there are microorganisms that, for instance, produce antibiotics or antimicrobial compounds that could be effective against pathogens. But there are also other microorganisms that can manipulate the immune system of the insects, protecting them from biotic stressors, namely predators, parasitoids, and also pathogens. Finally, we have also other microorganisms that can confer insecticide resistance to the insects. and also influence the insect behavior of the insects. They can also, only to give you an idea, there are microorganisms that can change the color of the body of the insect in order to avoid to be recognized by predators or parasitoids, and can also have an important influence by extending their longevity. So as you can see here, the microorganisms associated to insects, and in particularly to the insects that feed exclusively from xylem, are really very important, and their presence or absence could affect not only the fitness of the insect, but also their capacity to transmit pathogens, or inclusively their survival. So, taking this into account, we have tried to explore these microorganisms that are associated to insect vectors of Xylella, in order to use this knowledge to develop new tools, new sustainable tools to manage Xylella fastidiosa via their insect vectors. In order to reach this objective, we started to study the diversity of microorganisms that colonize the whole insect body of Philaeanus spumarius. So we started to work particularly with Philaeanus spumarius because it's one of the main vectors of Xylella in Europe. And so we started to access the microbial composition associated to the adults of this Philaeanus. Then we also try to see or to access the factors that can contribute for their shaping, and in particularly we have trying to see if there are differences in microbial composition among the different organs of the Philaeanus spumarius, and if there are differences among males and females. Then, based on these results, we are trying to see if this microbial community associated to adults of Philaeanus, if they provide advantages to the insect. We try to identify among the different microorganisms which one is important for the fitness of the insect. And then finally, we want to explore these microorganisms in the management, as I told you before, in the management of Xylella fastidiosa via their insect vectors. So I will show the results that we get so far, some preliminary results, and I will try to answer all these questions in order to reach our main aim, that is try to find sustainable tools to manage Xylella fastidiosa. So to assess the microbial community associated to the adults of Philaeanus, we started to study the bacterial community. For this, we have collected adults from the field, males and females. After the collection, we have surfaced these adults, and then under sterilized conditions, we have made the dissection of the different organs by separating genitalia from the head and abdomen. Then we have analyzed the bacterial composition of these three organs by using a metabarcoding approach. The results that we get, it was really surprising, because we observe indeed a great diversity of bacteria associated to the adults. So in total, we found more than 7,000 amplicon ASVs belonging to more than 100 genera, 65 families, 48 orders, and belonging to 9 phyla. The most dominant ASVs belongs to the phylum Proteobacteria, followed by Bacteroidetes and Actinobacteria. In terms of genera, we found that the greatest abundance belongs to Candidatus sulcia. So Candidatus sulcia, it encompasses several members that are obligated endosymbionts. That these endosymbionts, they colonize in particularly the domain of the insects, okay, in the specialized cells. These endosymbionts, they are very important for the insects because they provide the host with essential amino acids. Okay, so it is really very nice to find that this endosymbiont was one of the main genus associated to Philaeanus primarius. And then we found the second most abundant genus was Cutibacterium. This genus is also, was already reported in other insects, including in Philaeanus primarius. However, the role played by this genus in what concerns the fitness of the insect is so far unknown. So this genus is widely present in other insects as well as an endophyte in several plants, but the role played by these microorganisms are unknown. But we hypothesize that if it is present in high abundance in Philaeanus primarius, it means that maybe they play an important role for the insect host. And this is the open questions that we must also try to address in the future. In terms of diversity, so we found that we analyzed this diversity by calculating the Shannon-Weiner index, and we have compared the diversity among sex and among organs. And we saw that there are significant difference in terms of diversity among sex. So the males show significantly higher diversity comparatively to the females, and in contrast, we didn't found any significant differences among the different organs. So the abdomen, the head, and genitalia exhibit similar diversity of bacteria. However, when we compare the different sex for each organ, okay, we saw that the males show a higher diversity, mostly in the abdomen, in the head, and in contrast, the females show a higher diversity of bacteria in genitalia in comparison to the males. So there are, in fact, differences among in the diversity among females and males, but these differences depending of the organ of the insect. In terms of composition, so we have performed a Permanova analysis to see or to access the contribution of the organ and the sex for the composition of the final composition of the bacterial colonizing the Philaeanus primarius. And we saw that the organ and was really very important. It was a factor really important in the shaping of the bacterial composition in the Philaeanus primarius. So the organ itself, they explain about 26% of the bacterial composition variation in Philaeanus primarius. In what concerns the sex, so the sex was also found to be an important factor, but with a minor impact or a minor effect in comparison to the organ, contributing to 40% of the bacteria variation, composition variation in Philaeanus primarius. The impact of the organ in sex in bacterial composition variation was corroborated when we analyze a anometric multidimensional scale. So in this analysis, basically, we are trying to compare the similarity in bacterial composition among the different organs that are present, that are shown in this figure by different colors. So the abdomen in red, genitalia in blue, and head in green. And also to compare the similarity in bacterial composition in terms of the sex that are shown here by using different symbols. So in this analysis, we can see clearly that the different organs and the different sex, they cluster, they are separated between each other. And we can see that the differences or the bacterial composition of the abdomen, it's different, in particular the abdomen is different from the genitalia. So they are the most different ones. In contrast, the bacterial composition of the head showed to be similar to the abdomen and similar to the genitalia. In what concerns the sex, the greatest differences in terms of bacterial composition was found at the level of the abdomen, okay? And it was in the abdomen that we found greatest differences between males and females, followed by the head. So, based on these results, maybe we can see that these differences of bacterial composition among organ and sex, it means that maybe they are colonized by bacteria that could play essential roles for the insect. The differences that we found among sex could be probably due to different differences on physiology between females and males. And in terms, it's maybe, so we think that the role played by these microorganisms in the different organs could be different, okay? So, but this is our only hypothesis that we need to address, of course, in the future. So, since we found differences in the bacterial composition among the different sexes and organs, we have tried to see what kind of difference are these. So we, in this figure, we show the relative abundance of the different genomes among the different sex and organs. And we can see here that when compare the two sexes, the males are dominated particularly by Cutibacterium, while females are colonized mostly by the obligated endosymbiont Candidatus sucia. In terms of organs, we can see also clear differences among the different organs. For instance, the abdomen and the head, they are colonized mostly by Candidatus sucia, so this is an endosymbiont. And in contrast, genitalia was dominated mostly by Cutibacteria. The bacteria communities also differ, it was also different for each organ among sex. So as you can see here, we can see here, for instance, that when we compare the abdomen between males and females, we can see clear here that in the females there are a more abundance of Candidatus sucius in comparison to the males. The same for the head. When you compare the head of females and males, we can see clear here that the females show a higher abundance of Candidatus sucia, and in contrast, males show a greater abundance of Cutibacteria. So these results also corroborate that there are in fact differences among sex and organ, and maybe these differences could indicate that the bacteria that are colonizing specifically males or females or different organs could play an important role for the biology of the Philaeonus spumarius. Then we have also analyzed in particularly the symbiotic genera. So we based it on the literature. We try to identify the symbiotic bacteria that are already described in the literature. And the results show that 3 genus, namely Candidatus sulcia, Allomonas, and Sodalis, were found in all the organs and sex of Philaenus. So it means that maybe they are crucial for the survival of the Philaenus because they are present in high abundance and also present in both sex and in all the organs. Although these results, we found that there are other genera that appear exclusively to a particular sex or to a particular organ. For instances, we found that Flavobacterium and Streptomyces were exclusively present in females, and in contrast, Citrobacter and Rhodococcus were found exclusively in males. In what concerns the organs, Citrobacter, Flavobacterium, Rhodococcus were exclusively found on the genitalia. So based on these results, we hypothesize that the exclusive presence of these genus to a particular sex or to a particular organ could play an important role or an important function for the insect. And to corroborate this hypothesis, we perform specificity analysis in order to find families, bacterial families, that exhibit significant specificity to a particular organ or to a particular sex. The results are present in this figure, and we can see that the organ show or exhibit a higher number of genus that are specific than to the sex. In what concerns the sex, we found that only a single family shows specificity to the sex, and particularly this family. And we found that although this family are present in both sexes, they show higher abundance in females. In what concerns the organ, so we found 7 families that show a strong specificity to the organ. And among these 7 families, one of them, Microbacteriaceae, exhibit the strongest specificity to the organ. And this family was shown to colonize or to be present in higher abundance in genitalia in comparison to the head or to the abdomen. So the specificity in particularly of these bacteria could reflect different functions played by these microorganisms in that specific organ. For instances, in what concerns the organ, we show that the Microbacteriaceae was specificity to the organ, and it appears in higher abundance in genitalia. This family encompasses several genus that we have detected in our work, namely Curtobacterium, Agrococcus. So all these bacteria, all these genus belong to this family, and we found that we detected to be associated to Philaenus. And we hypothesize that these bacteria may be relevant to the reproduction system of Philaenus spumarius, okay, because they are in particularly present in the genitalia. In what concerns the sex, so we found one family that was highly specific to the sex, and this family encompasses one endosymbiont, Candidatus sulcia, that it was found in great abundance, in particular in females, And since Candidatus sucia, it's an endosymbiont that it's known to provide the host with essential nutrients, in particular amino acids. So it is likely that the Candidatus is more crucial for females' nutrition than for males, because we found that this genus in great abundance in females when compared to males. So we hypothesize that this genus could be more important for females' nutrition than in males. So maybe males, they use other microorganisms to supply them with these essential nutrients, namely amino acids. So besides these genus, we have also found other genus reported in the literature as symbionts. And among the symbionts identified in our work, we found some of them that are described to manipulate the insect reproduction via male killing or parthenogenesis, namely Rictes and Flavobacterium. But we have also found other symbiotic bacteria that are described to be important by supplying essential nutrients that insect doesn't have by feeding in the xylem, namely amino acids or vitamins. Also, endosymbionts identified in our work are known to provide insecticide resistance, others to fix nitrogen within the insect bodies, others to produce antibiotics, and finally, others endosymbionts such as Seraken, Providence, and Sodalis that are described to protect the insects against biotic stress, namely parasitoids. But so these endosymbionts in Philaenos spumarius could provide the same protection to the Philaenos. Okay. So finally, we have tried to see if some of these bacteria that we have identified could be useful for the Philaenos spumarius. And to reach this objective, we started to do a preliminary work by using a model insect such as Drosophila melanogaster. And what we did was select 5 bacteria, because I didn't mention before, but we have access to diversity of these bacteria by using metagenomic analysis, but also using cultivaval methods, the cultivaval approach. And from these cultivaval microorganisms, we have selected 5 to see their effect on some morphological and parameters, and on the capacity to inhibit the production or to reduce the progeny of this model insect that is Drosophila melanogaster. So what we did was we selected these 5 bacteria isolated from Philaenos spumarius. We have inoculated these bacteria in the food of Drosophila melanogaster. We cultivated the Drosophila melanogaster in these flasks with the food and inoculated with the bacteria and the respective control, of course. And then we have evaluated 3 generations of this the progeny of the Drosophila melanogaster. For each progeny, we have analyzed the total number of pupae, the number of adults, either males and females, the number of the larvae, and some morphological parameters of the adults, namely the size of the body and the wings of adults. So the results are very diverse according to the microorganisms. So here I show you only a summary of the results. So the microorganisms are shown here by codes and by different colors. And as you can see here, some of them, they show positive and they have some negative effects, and others have positive effects on the different parameters evaluated. For instances, in the first generation, we saw that the microorganisms B27, they promote the number of pupae, the number of adults, they increase the size, they reduce the size of the body of the males and females, and they also increase the death of the larvae. So as you can see here, the results are very different according to the different isolates that we use it to inoculate the food of Drosophila melanogaster. A result that we saw is that the effect of the inoculation of the food with these microorganisms can have impact over the different generations. In some cases, the impact, the negative or positive impact increase, and in other cases reduce or disappear depending on the microorganisms, okay? So what we wish with these results is that after the knowledge, the presence of these endosymbionts in the Philaenus spumarius and also their potential role, our main aim is to try to use this knowledge that we get from this study to develop new tools to manage the Xylella fastidiosa via the manipulation of the microbiota of Philaenus spumarius. So this manipulation of the microbiome, the main aim of the manipulation of the microbiome is to have in a way that can reduce directly or inhibit directly the pathogen in the foregut of the Philaenus spumarius, or the manipulation of this microbiome can also have an impact by reducing the capacity of the vector to transmit Xylella fastidiosa. So to reach these objectives, we have already available different approaches. For instance, the paratransgenic-based approaches allow us to use these symbiotic bacteria. So we identify these bacteria that are well established in insects, and we know that can be transmitted to the next generation. So we select these bacteria, and then we try to engineer these bacteria in order to express molecules, effector molecules that have a harmful effect directly in the pathogen. These symbiotic engineering bacteria can be inoculated in Philaenus spumarius, and then this Philaenus spumarius can be released and express these molecules that have an harmful impact in the pathogen. Another approach that we can use it's incompatible inset technique. So the incompatible inset technique, it's based on the cytoplasmic incompatibility, the best approach or the best known use of this technique is by using Wolbachia. So Wolbachia, it's an endosymbiont that it's transmitted exclusively by females. So the males can be colonized but cannot transmit it to the progeny, this bacteria. And Wolbachia is an endosymbiont, very important for the fitness and for the survival of the insects, because they manipulate the reproduction of the insects. And in this process of the cytoplasmic incompatibility, the males that are infected by this endosymbiont, when mate with non-inoculated females, we will not have offspring. So this process, we can use this approach to reduce the population of Philaeanus spumarius in the field by infecting males and by releasing, in this case, uninfected females. So we can in the lab remove or these endosymbionts by using, for instance, antimicrobial compounds such as peptides, etc. And this can this can make the females sterile, and we can release it to the field. And when they met with the infected males, they will not be able to produce progeny. So these are in fact different approaches. Different approaches that we can use it, but we are far from to use it. Okay, so we have already some techniques, basically omic-based techniques, that allow us to better know the presence and the composition of this microbial community that are associated to Philaeanus spumarius and other insect vectors. However, there are still some challenges that we need to overcome and that need to be addressed. Namely, we need to generate more knowledge of the relationship between these endosymbionts present in the vectors, okay, the plants, and also the pathogen. And we need also to develop new tools that allow us to manipulate these microbiome that are associated to the vectors, namely Philaeanus spumarius, in order to reduce the survival of the pathogen and also the capacity of the insect to transmit its Xylella fastidiosa. So it's a long process that we need to overcome. So finally, I would like to thank, because the work that I present here was done mostly by Cristina Camerão. So it's a PhD student that her work is devoted to the study of the microbiome of Philaeanus spumarius and to manipulate this microbiome for the management of Xylella fastidiosa, and also to the group of the Mountain Research Center and the different projects that funded the results that I show you here. And thank you very much, and I'm able for questions.
Thank you, Paula. Thank you. I think it has been a very, very detailed presentation on the microbiome of the vectors of Xylella, specifically on Philaeanus spumarius. And although until the moment there's no questions as in the previous presentation, it has been very, very interesting to me, and I have some questions to you. First of all, possibly it's a lack of knowledge from me about the subject, but all the studies on the effect of the presence or absence of this bacteria on Philaeanus spumarius have been done all the assays not on Philaeanus.
So
so
if I understood correctly, so you are asking if we are assessing the diversity, the composition of the bacteria in Philaeanus. Yes, so the methods that we use it allow us only to know the presence or absence of a specific microorganisms, in particular bacteria, because we only have surveyed the bacteria in the adults. Yeah.
But all the essays you have shown on, for example, mosquitoes, you have not done this on Philaenus, possibly because it's difficult that the origin of Philaenus, of course, possibly for that.
Yes, yes. So we only detect the presence and absence, and based it on the literature, we try to see the role that these microorganisms could play in the Philaenus spumarius. So it was only based, the ecological function of these microorganisms are based on the literature. Okay, so we have only performed some essays by inoculating Drosophila melanogaster. So we use it as a model organism because it's more easy for several reasons in comparison to Philaenus spumarius. So it's more easily rare in the lab, etc. So, and we use it Drosophila melanogaster in order to elucidate the rule of some of the bacteria that we isolated from Philaenus spumarius. So this is the only data that we have regarding the function rule of our microorganisms, because so far we have only the majority of our data is only related with the presence and absence of particular bacteria.
Okay. We have a question from Francesco Volpe. He said, can the Longovarian parapause have an influence on the result zone depending on the time of sampling?
Okay, so could you repeat?
Yes, can the Longovarian parapause have an influence on the result zone depending on the time of sampling?
Yes, I believe that it could have. So I don't have data to answer to be sure. Okay, so we never studied this, but I believe that depending of the period of the year, depending inclusively of the host plant used by Philaenus spumarius, because it was interesting results that we have that some of the microorganisms that are colonizing the Philaenus spumarius are already reported as endophytes of plants. So we believe that most of these microorganisms that are present in Philaenus are acquired from the host plants, from the plants that they feed. So I believe that all these factors, the time of year, the plant that Philaenus used to feed on, etc., could have influence in the final composition, in the final bacterial composition of the Philaenus spumarius.
And a question, I have another question to you regarding this, the importance of this bacteria on Philaenus spumarius, at least. You know if there are research groups currently working on the presence of these bacteria in different populations of Philaenus spumarius around the world? And do you think this is a necessary activity to continue the work?
Yes, of course that it's really important to know, because I think that in the future we will look more deeply for the microbiome, not only of the insect, but also from the microbiome of the plants, and in order to try to explore, to manipulate this microbiome in order to do the management of diseases or pests. And in what concerns in particularly the exploitation of the microbiome of insect vectors, of mosquitoes, or for instances, or including in case of Philaenus spumarius, I think that we are only in the beginning, because we know already, and we have already techniques that allow us to know very well the composition of the bacteria that are present, their function, etc. However, the way how we can apply this knowledge to do the management of the pest, or the management of the pathogen via the insect vector, it's more difficult. So, and it's because I think that it is really important to innovate in terms of new tools in order to reach the manipulation of this microbiome, to reduce not only the pathogen survival in the insect vector, but also to reduce their ability or their capacity to transmit the pathogen. I gave you two examples, two different approaches that we can use it, but when we try to apply them in the field, so it will be more difficult. Okay, so I know that this area, it's more developed, it's more advanced in terms of the mosquito, but the results get so far are not so promising. Okay, because it's very difficult to put it to reach the effective impact by using these approaches in the field. We can, another approach that for the manipulation that I didn't mention in my presentation, but we can also try to use antimicrobials to change or to manipulate the microbiome of the insect by using natural compounds, not only antibiotics, because we want to reduce the use of antibiotics, of course, but by using, for instance, peptides or other kind of natural molecules that exhibit antimicrobial activity. And by using this approach, we can also manipulate the bacteria that are colonizing these insects. So this is another approach, but of course that it's a long process to put it in the field. Okay.
Thank you. It's to me it's a really very attractive line of research, and I'm okay with you. Okay, thanks, and it's all for the moment. Thank you.
Thank you very much.
So now, then we are going to listen the third speaker of the day, who is João Spoti Lopes. João is professor in the Department of Entomology and Ecology of the School of Agriculture Luiz de Queiroz in Sao Paulo, Brazil. In 1994, he established the Laboratory of Insect Vectors of Phytopathogens and has dedicated his research to the study of the transmission of phytopathogens by insects, ecological, behavioral, and molecular studies of pathogen-plant vector interactions involved in the dissemination of phytopathogenic bacteria and viruses, aiming to obtain information about pathosystems for the effective control of diseases in cultivated plants. I think it's all this, no? So today he's going to tell us about the vectors of Xylella fastidiosa in Brazil and their ability to transmit the bacteria. Okay, João, it's your turn.
Okay, can you see? Good afternoon, Paco and other friends, other colleagues. Can you hear me and see the screen well?
Yeah, it's okay.
Okay, thank you. Yes, I try to move the screen here too, so that I can. Okay, nice. Everything is okay, huh? Okay. So first of all, thanks. Thank you very much for José Pereira and other coordinators of this workshop online webinar for inviting me for this presentation. It's a pleasure to talk about Xylella fastidiosa in the vectors, a subject that I have been studying for several years in Brazil, and it's a very exciting field of research. The idea here is to give, after these two very nice presentations by Paula and by Blanca, is to give an overview of vectors of Xylella, a little bit of transmission biology, but very synthetic, and explore a little bit of the information we have on vectors in Brazil associated with our fruit crops and major diseases, and more importantly, to have an idea of their importance and how we determine for the major diseases that we have, what are the vectors, a little bit about the transmission biology, and also about their relevance in terms of epidemiology. And a lot of this importance comes from ecological and behavioral characteristics that these insects have. And this is the basis also for managing strategies. So a lot of the strategies come from the understanding of the epidemiology and also about the vectors and where they come from, where they feed plants and habits and so on. So this will be the topics of this talk. I'd like to acknowledge also some funding agencies that have helped us to do research. CAPES from Brazil, FAPESP, our State Research Foundation, CNPq, which is our federal state foundation. Also, we have support from European Union. I have been participating with the European group since Horizon 2020, and now I work with the vaccine too. So it's been very, very, very good to work with all the team. And also the work I'm presenting here, most is about the work we are doing here in Brazil, but also with collaborators in other countries. So before we enter, it's important to point out that Xylella fastidiosa pathosystem is highly complex. So you have, as Blanca showed, a lot of genetic variation within Xylella in subspecies and sequence types. We have lots of host plants, although in a particular situation, like in a pathosystem, in a given region, you may have a much more limited number of hosts, but sometimes there are several hosts, and many are asymptomatic, so we need to know their importance. And my focus here will be on the vectors, which are also polyphagous and feed on several plants, so some of them can be really important. Abiotic factors can also influence everything because you have different climate, sometimes practices like irrigation that modifies the vegetation, even in the underground and also in the surrounding vegetation, and that will modify also the vectors and the pathogen involved. Okay, so in Brazil and in South America, we have here three major diseases associated with Xylella fastidiosa. Citrus variegated chlorosis affecting citrus, coffee leaf spot, plum leaf scald, and now more recently, olive leaf desiccation. I will comment on the four very quickly, but I will focus on CVC to show how we use the information of vectors, ecological and behavioral, to determine the main vectors and how we integrate these into management strategies. So in South America, we have this. The first disease detected was plum leaf scald, was detected and reported in Argentina in the 1930s, and then in southern Brazil in 1978 by Kitajima and French, and in that report they associated the disease with this bacterium. The bacterium shows in plants very strong symptoms. You see the leaf scorch, but the plant is very susceptible depending on the cultivars. At that time, most of the cultivars were very susceptible, and you see the death of the branches and then of the tree in a few years. And it's been a very limiting factor of cultivation of plants, especially in São Paulo and Minas Gerais, warmer regions where the bacteria is very severe. But Xylella fastidiosa became really important in Brazil after it attacked a major crop, a very important crop like citrus. Citrus is in São Paulo state is really important crop. São Paulo produces about 60% of the orange juice in the world, so it's a big business here. A lot of people employed, a lot of revenue exports. So when it reached citrus, then it became really relevant. In the beginning, you see this leaf chlorosis, which has a variegated parent. The infected plant is not killed, but you see a severe stunting and some dieback of the branches, apical branches, and you see a reduction in fruit size. And this is a kind of symptoms on the underside of the leaf. You see lesions, which is very characteristic and diagnostic of Xylella. So sometimes you see some deficiencies that have these yellow spots, but the diagnosis is based on these gummy lesions on the underside of the leaves in the corresponding areas on the upper side. So this was detected in the late 1980s, but it was demonstrated the association with Xylella in 1990. In 1993, it was the Cox postulates were concluded. People started looking for other hosts of Xylella nearby citrus, and they found that coffee was infected by Xylella as well, showing small fruits, shortening of internodes, in a disease that was called coffee leaf scort, because you used to see some scort at the apical of leaves, and also branch atrophy because of the leaf drop and reduction of the internodes. Later, with the molecular characterization of the strains, and also biological characterization, we learned that the isolates are different from citrus. At the beginning, they thought were the same, but now we know that they are genetic and biological different, although they belong to the same subspecies Palka. CDC became also important because it was rapidly spread to other regions in Brazil. In São Paulo, it was already epidemic in 1994, but it reached several states because São Paulo is a major nursery tree producer. So it exports nursery trees to several places in Brazil. In the past, it used to be like this. And the nursery trees were unprotected, so they were contaminated with the bacteria. So that's the way the bacterium reached other states distantly located from São Paulo in a few years. Today, all citrus regions in Brazil have Xylella, including the Amazon. We have groups now working in Xylella in Manaus, in the middle of the Amazon forest, because they have citrus growers there, and they are finding the bacterium. Also in Argentina and Paraguay. More recently, the Xylella was found to infect olive trees too in Brazil. And also in Argentina. In Argentina, there was a report in 2015, and in Brazil, Elvésio and other colleagues, we detected this bacterium also infected olives and associated with this branch desiccation, leaf desiccation that you see, especially in the Mantiqueira mountain region. Where olives are grown in São Paulo and Minas Gerais, because we are like in a tropical area. So we need to grow the olives in high elevations, above 1,200 meters, where the temperature is lower, and you have fruits. And here in this region, it's a very hilly region. So you have several small orchards scattered through the mountains, And in 83% of the orchards, in a survey done in 2020, we could detect Xylella. And most of the symptomatic plants, it was they were positive for Xylella fastidiosa. So it's a kind of important disease in that region. We start working in 2015, sampling sharpshooter in these orchards to determine the major vectors. At the beginning, the plants were healthy. And 5 years later, when we were finishing our service, some of these orchards were already dead. So it's a very severe disease. A lot of growers are having problems, and some quitting planting olives because of this. So this is a sad story in Brazil as well. Transmission of Xylella can occur by grafting, by infected buds. And that's how they do it. When they produce nursery trees, they have to have a sign put on a rootstock. And if the mother tree is infected, that can be transmitted with the sign. You can also have root grafting. This was experimentally demonstrated, this type of transmission. But in the field, once you have infected plants, the spread is done by vectors, by insect vectors, because it's a xylem-limited bacteria. It depends on vectors to acquire and transmit them to other plants. The vectors, because the bacteria is xylem-limited, the vectors are insects that have the ability to feed in the xylem. And we call them xylem sap feeders. You have several groups of xylem sap feeders in Alkenorhyncha, including the Cicadelids. There's a subfamily Cicadelinae, the so-called sharpshooter leafhoppers. So the Cicadelids are the leafhoppers, but the leafhoppers of this subfamily Cicadelinae, they are called sharpshooters because of their habit of hiding themselves in branches, in leaves, when they perceive somebody. We have two tribes, Proconiini and Cicadelini, and we have the hundreds of species in each of these tribes. Another group in Alkenorhyncha that has xylem sap feeders is the Cercopoidea, the superfamily with different families like Aphrophoridae, Cercopidae, and Castopteridae, with vectors that also contain vectors. So these are all xylem sap feeders, and we can have species that are important in different parts of the world. In Europe, for example, the Aphrophoridae, like Philenos and some Cercopids, are quite common, so they are generally much more important than the sharpshooters. Whereas in United States and Central America and Latin America, sharpshooters are very, very abundant and usually more important. Okay, now that we know a little bit about the group, let's talk about the transmission process. Just for you, for those that are not familiar with Xylella and have an idea of how the bacteria is transmitted. So the transmission process has basically three major steps: acquisition, when the insect feeds on infected plant; retention, in the foregut, the bacteria is retained in the anterior part of the stomach, of the gut, and then from there it's detached and inoculated into health plants. So these are the three major steps of the transmission process. Here, so the insect, for acquisition, it has to feed on the xylem vessels. When it starts ingesting the sap, the bacteria is uptaking together sap, and it will adhere to the cuticle of the foregut, especially the presiberium and the siberium, where it's indicated here by this red material. And here we have this nice drawing made by Eleni Bacchus from USDA. It shows this, the anterior part, the anatomy of the anterior part of the gut with this suction pump. And also you see here the styletes, the maxillary and mandibulary styletes, they are coupled together and they form the food canal and the salivary canal. And these styletes, they are held by this, this labium, which is a kind of a case for the labium. So everything here is removed to show. And here you see a duct that connects the food canal from the styletes to the cibarium. It's called the precibarium. And this is one of the places where, and I think we believe it's the most important retention site of the bacteria. This is very narrow duct that connects the stylate food canal with this cibarium chamber, which this kind of a wine glass shaped structure that we have here. This chamber has a decreased volume when the muscles are elongated here, but when they are contracted, this cibarium dilation muscles are contracted, they open. This chamber increases in size and sucks the xylem sap under negative pressure in the plant. And the bacteria is uptaken and will stick somewhere in this area here. So this process, the acquisition, can occur as soon as the insect reaches an infected xylem vessel, and it will attach. Later on, the insect, when it moves to a healthy plant, it can inoculate it. Experiments that are done by allowing several minutes and 1 hour for acquisition, just taking the insect and put it on a health plant, they can transmit after 1 hour, for example, on a healthy plant, they can already transmit to a health plant. So basically, there is no measurable latent period between acquisition and inoculation. Of course, if you have a longer acquisition access period, you have a higher efficiency, as I will tell later, but there is no measurable latent period. Also, there is no transmission after molting. If the bacteria is acquired by a nymph feeding on an infected plant, and if this nymph undergoes an ecdysis, where the exoskeleton is removed, the cuticular lining of the foregut is removed as well, because it's composed by this exoskeleton, and the bacterium is lost. So transmission is not retained after a molt by nymphs. But if the acquisition occurs in the adult stage, then the insects can retain this ability to transmit for several days. This is a study done a long time ago by Hill and Purcell, and they showed transmission up to 60 days with, in grapes, for example, showing these persistent characteristics of the transmission. So here it's just a summary of this transmission characteristics. So acquisition, inoculation, minute and hours is the duration. There is no latent period in the vector, no transstadial transmission, no transovarial transmission too, which are characteristics of a non-circulated pathogen. So it's foregut-borne. All the transmissible bacteria is retained to the foregut. If the foregut lining is removed or lost, for example, by a nymph, the bacteria loses the vector loses the ability to transmit. Persistent transmission, as I mentioned, by adults, and the bacteria is also propagated. So the bacteria, after it adheres to the cibarium, is able to propagate and actually forms a biofilm with lots of cells forming a kind of a carpet of bacteria. These are classic pictures from studies of Purcell and Berlusky. But this same type of pictures has been obtained in other studies, including here in Brazil, showing similar characteristics of colonization of mouth parts. So here is the cibarium, the wine glass shaped structure. In this, as you see, some bacteria is retaining here, but there is a lot of bacteria here in the presiberial channel, especially close to the presiberial valve, which is a valve that closes and opens this duct. And by doing this, the insects control the movement of sap. And there is also some turbulence, and this may dislodge some cells that can be maybe returned to the plant. So there is a biofilm formation in the insect. First, the bacteria has to be adhered to the surface. This is a model that was developed by Chiellini, Almeida, and others have contributed to. Basically, there is an initial attachment mediated by afimbrial carbohydrate binding proteins. They are initially bound laterally to the cuticular lining of the foregut, and then there you have cell-to-cell adhesion, and then they form these microcolonies, as you see here in this scanning or in this electron microscopy pictures. Then they become polarly attached as the biofilm matures, and then you start seeing some cell division, and some cells are detaching. So this is the evolution of the biofilm in the vector with propagation of the bacteria. That's why the vector retains for long periods of time. And you see here the model showing the detachment and release of bacterial cells that are supposedly released and inoculated into plants. So these are mediated by proteins present, this attachment by proteins present in the membrane of the Xylella fastidiosa, and also by fimbrial structures like pili that they can help, especially in the polar attachment. They are thought to do so. Inoculation is a third step of the process. This is the least understood process. So in the recent years, researchers have been dedicating more time to understanding this process. Colleagues like Alberto Ferreres, Rodrigo Almeida, Cornara have doing these studies using EPG, together with also Elaine Barcos. Lots of information have arrived, but what we know is that foregut colonization, full colonization is not necessarily required, for example, because you don't have a latent period. So before a very strong biofilm forms, the insect can already transmit. But it has also been observed that if you have higher populations, like in Philenos, you get a higher transmission rate too. Inoculation is presumably related to egestion, which is the return of liquids from the mouth parts and from stomodel to the plant, regulated by that presiberial opening and closing and fluttering that creates a kind of a fluid flow dynamics that may facilitate the cell detachments. Transmission efficiency, ability to transmit, is influenced by various factors and interactions among factors. So you may have different transmission rates depending on vector species, Xylella fastidiosa genotype, host plants, temperature, also duration of acquisition access, inoculation access period. So it's usually the longer you allow the insect to feed on a plant, up to maybe 2 or 4 days, you get higher transmission efficiency. Populations of the bacteria in the host plants is a very critical factor. So usually if you have hosts with higher bacterial titers, as a general rule, you have a higher transmission rate to test plants. And you may have other more interesting factors like differences in populations within a given host plants may interfere with acquisition efficiency, depending on the vector behavior. Sometimes the vector prefer to feed on a tissue that is on organ that has a lower titer, so it will be less efficient than the vector that feed on higher titer organs. And also disease symptoms, the severity of disease also has an impact on the transmission efficiency. For example, we did a study here in Brazil. A student of mine, Rosângela Marucci, she compared the preference of the insects when they were offered plants showing severe symptoms and health plants, or plants showing very light or that were asymptomatic plants, infected but asymptomatic. So usually in the beginning, the insects do not discriminate well in the first hours, but when they start probing the plants, they don't like it, so they move to other plants. So there is a kind of behavior that they usually avoid plants that are very sick, and they tend to stay longer in plants that are healthy or that have no obvious symptoms. Source plant species, there are several studies that show this, the effect of source plants. For example, Xylella fastidiosa subspecies, isolates of these subspecies, they can infect usually grape, but not only grape, but also almond and alfalfa. And here there are a study done with alfalfa and isolates from alfalfa. It could be it's acquired with and transmitted with much higher efficiency than when the same isolate is infecting almond and grapes by this particular vector, like Draculacephala minerva. So Draculacephala minerva, the green sharpshooter, it likes alfalfa, so it feeds well, especially on parts of the plant that have higher population. So it's a good vector in alfalfa. But when it feeds an almond, infected almonds and grape, transmission efficiency is very low, showing that this interaction between the vector and the host plant is very important for the transmission success. We have done studies here investigating transmission efficiency between different species for a single sequence type, for example, using this membrane feeding technique that allows us to do the acquisition on cultured cells. So by doing this, we eliminate the source plant effect. It's a major effect because we have vector behavior. Some species like one host plant, the others don't like the same host plant. So in order to remove this behavioral aspect, we offer the bacterium in this membrane sachets and did the study. So this is done with this nice system that was developed in Berkeley by Nabil Quilini and Rodrigo Almeida. And so they developed a good medium for doing this. And you can put a droplet of cell suspension in this kind of a chamber, a feeding chamber, which has a membrane sachet with the cultured cells in a diet, an artificial diet that insects feeds. We allow the insects to feed for like 6 hours, 8 hours in this situation, and then we remove the bugs and place them on test plants. And then we measure the transmission efficiency from this. So we did this for different species. These are vectors in citrus, and you see here the dark bars is the acquisition rate, and the empty bars or white bars are the transmission rates. So you see that there's no significant difference in the acquisition rate as we expect, because they are feeding on the same cultured cell suspension. But we do see some differences in transmission efficiency by different species of vectors. We also we fixed the vector species and changed the sequence types of the Xylella fastidiosa and offered these different sequence types in membrane sachets for the same vectors. And we did the similar measurements of acquisition rate and also transmission rate. Here, as you see, the transmission rate, the acquisition rate in dark bars, there is there is only one citrus type that was acquired at lower rates. And concerning transmission rate, one of them was transmitted with higher efficiency. But in general, all of them are transmitted by the same sharpshooter species. So, as a general rule, as it has been stated in several reviews about this subject, we don't have like a transmission specificity. Yeah, like any insect that is a xylem feeder is a potential vector. This was stated, it was known since the 60s, and work by Fraser. He stated that after testing several species of sharpshooters and also some sarcopods, I think they found this that's all of them were transmitted. But one question is, should we be concerned with all potential vectors? Because if any xylem sap feeder is a vector, then we have problems in regions where we have many vectors, especially in the tropics. So the tropic nightmare is that we have lots of species. We have, for example, in citrus, over 30 species of sharpshooters and spittlebugs. Coffee, almost 40 species. In olives, more than 100 species altogether, sharpshooters and spittlebugs were recorded. So it's kind of a concern if all of these bugs were considered relevant. So we have to consider here the concept of a vector and this vector or the vector. So a vector is any species that can transmit under certain conditions. The vector are vectors that are really important epidemiologically, and we should, these species, we should focus for managing and practice and so on. So how we determine that a vector is relevant, the vector? We usually consider ecological characteristics like population density, mobility, natural infectivity, transmission efficiency, host plants, the host plant associations of these vectors, if they feed on plants that are hosts of the bacterium. And also we have to consider the epidemiology, especially the inoculum sources and types of spread. So I'm going to show this using CBC as a case study. So we have this well-known disease in Brazil where lots of work on ecology of the vectors has been done. So we know well the information. Okay, so in citrus, as I just mentioned, we have several genera. We have 29 genera of xylems feeding Alcanorhynca, and they are all potential vectors because these were samplings carried out in citrus orchards. Experimentally, 13 species, almost half of them, were shown to transmit the bacteria. And most of them were classified in citrus as predominant species by faunistic analysis. Transmission efficiency to citrus is variable. Some of them are very inefficient, like Acrogonia virescens, Homologisca, for example, and whereas some are moderate efficiently, like Macrogonaria, Bucephalorgonia, Lobopterus, are a little bit more efficient. So transmission efficiency is a parameter. So we could tell based on this data that some are more efficient than others, but we should also consider the presence of the bacteria in the field. So are these species natural carriers of the bacteria? If they are natural carriers, it means that they feed on plants that are inoculum sources of the bacteria. Maybe citrus plants or maybe other plants present in the underground vegetation I mean, in the ground vegetation and also in the surrounding vegetation. Here you see data of some species that were shown to be infected by this bacterium, and others that were not shown to carry. Of course, this is a sampling. Huh? We may be losing. It's not easy to find the bacterium in the field, in the insect. So we have to sample a large number of individuals in order to be sure. For example, in order to ascertain that Cometopula facialis, for example, is not carrying. Probably it's carrying, but in the sample we did not get any of this. Concerning the location in the orchard, so do they feed on citrus plants or do they feed only in the spontaneous ground vegetation? So this is important too, and this has been determined. Some species like Cometopula facialis, Acrogonia, Gilolopterus, Romalodisca, they are all found in the citrus canopy, whereas some are only grass feeders. They feed on the grasses, especially grasses that are predominant in the spontaneous vegetation, in the ground vegetation. And some species like Bucephalogonia xanthophytes, they occur in the ground vegetation and also in the canopy. So this species here, there are studies, biology studies that show that they lay eggs in the citrus trees and they feed on the citrus trees. Whereas those that are in the grass, sometimes they are found in the canopy, but they are very rare. Usually they stay only in the spontaneous vegetation. And then there is this question: are the grasses or other herbaceous plants' presence in the ground vegetation important in the epidemiology? So some studies have investigated the presence of the bacteria in these grasses by injection and by natural collections. And they have found species that are infected with this bacteria. Some are quite frequent, like Echinocloa cruzgali, Brachiaria plantaginea. But when they try to use these infected plants as sources for citrus, the study showed no transmission. So we don't have evidence that they are sources of inoculum. And another consideration is that these grasses, they grow very fast, and they also mow it frequently. So the assumption is that because of the very fast cycle of the plant, the bacteria may not reach titers high enough for acquisition by the vectors and then transmission to citrus. So we think that they are not relevant, and also this is supported by the studies on epidemiology that consider the spatial patterns of disease and also temporal patterns that have shown that citrus is a major source of inoculum for CVC, if not the only source of inoculum in our conditions. We see both primary spread from neighboring orchards and secondary spread among plants within an orchard. And the infected citrus plants are the major drivers of these epidemics. So we believe that, and we consider this in management, that the key vectors for spread among all those 29 genera are these four guys: Acrogonia citrina, Gilobopterus costalimai, Oncometopula, and Bucephalogonia xanthophytes, especially because of their population density in orchards, but particularly for their ability to colonize citrus trees and move the bacteria from one citrus to another. And besides, they are confirmed vectors of the pathogens. So based on that, CVC management, based on this and other information, of course, CVC management has been established since the middle 90s in Brazil. basically with three major tactics. One is the production of healthy nursery trees. Other is roguing of infected plants, sometimes pruning, I will explain that, and also vector control. So for healthy nursery trees, the nursery tree production system in Brazil was completely transformed to be a protected system. It was originally in open fields, and by the late 1990s, Instituto Agronômico, along with other research institutions, the Embrapa and others, developed this, Fundecitrus developed this system where the plants are produced under protected conditions. Mother trees, the scions are healthy, and also the rootstocks, and the nursery tree is completely produced under this protected system. So the nursery tree is healthy and is planted healthy. So this was a mandatory action, and after it was implemented in São Paulo state, the incidence of infected plants in the field was dropped from 35 to about 30%, 7%. So it was a very important factor to reduce the sources of inoculum in the field. Together with this, of course, you have the eradication measurements like roguing and insect control. Young citrus groves from 0 to 3 years old, inspections should be done 1 to 2 times per year, and any plant that shows symptoms should be eradicated, because when the plants are young, the infection becomes systemic very, very fast. And insecticides are usually treatments are done very frequently in this age, because the plants are very susceptible. And if you get an infection early when the orchards are very young, then you have a inoculum in the field, and it's much more difficult to control the disease spread. In older groves, you still do the inspection of diseased plants, especially in the late summer and fall. That's the time when the symptoms become more obvious, which is in Brazil is from January to June, more or less. And plants that show level 1 of severity, level 1 indicates like that one branch, isolated branch, has a few leaves that are show that chlorosis, leaf chlorosis. So that indicates that the infection is starting, and you know the branch. Then you can prune that branch 1 meter below, and you may get rid of the infection and save the tree. But if the plant showing signals of systemic infection, if you see like symptomatic leaves in one branch and then the other and then the other, it means level 2, the infection is systemic, and then the plant has to be eradicated. But this is something that has worked only with citrus so far. Other host plants have been this type of strategy has been studied, including olives, and doesn't work. So the citrus growers, they have this option. They can save plants if they do inspections very constantly. Vector control in other groves can be based on surveys too. Insects, the yellow stick traps are is a very good sampling device. The insects are attracted to the yellow, and they can be hanging on the trees every 30 meters, 50 meters, especially in the edges of the field, because many of these vectors come from neighboring vegetation or neighboring groves. So they are trapped, and if you get higher populations, or when you detect the vectors, you can do the applications. Vector control, in most cases in Brazil, until recently, they were done with insecticides. And insecticides can be applied in the soil or in the trunk. And some of them have systemic action for young plants as long as they apply in the rain season. That gives a longer residual period. But if the trees are older than 3 years, that doesn't work well in Brazil. And then they will have more a contact action, and they will be more foliar applications, and which several groups can be used to control the leaf hoppers, neonicotinoids, pyrethroids, carbamates, and new groups that are available now. So these measurements, especially the planting of healthy trees, roguing, and use of insecticides, has worked and resulted in a very strong reduction of incidence in the last 10 years. As you see here, today is less than 1% in the last surveys. But this was not because the citrus growers were fighting against Xylella fastidiosa. This was because they were fighting against fungal bean, which is a much more devastating disease for the citrus grower. So in CVC, they were kind of lazy. They were not removing many trees. They were keeping the plant producing because the tree does not, there's no death of the tree. But fungal bean is a disease that really force the citrus growers to remove the plants. So a lot of plants with the citrus greening epidemics were removed, especially the older ones. And the vector control, in the case of citrus with insecticides, was done much more intensively, and that resulted in a very drastic reduction in incidence of this disease. So the methods work, but they have to be adopted. When they are adopted, you get good results in restraining the disease spread. Of course, insecticides a lot is not good. This is not sustainable. So right now, research is now seeking for sustainable approach. So we have here the usual methods, but we need for vectors to select other more biorational methods, not only selective insecticides. We have new insecticides that are more selective, but especially biological control agents. So there are studies we are doing, for example, under this new European project, looking at the effect of entomopathogenic fungi on the bacterial colonization and also on the insects. And also there are other things to be exploited, like natural enemies, egg parasitoids, for example, are very common, so it could be another option. Plus, there are interactions should be targeted, not only direct effects on the vector or on the plant, but also the interactions. So ionization through resistant mechanisms or other tactics. For example, there is the NAC methods, there is the more like a curative measurement, but also molecules like GSF that regulates cell-cell communication. So there are other strategies underway. I'm not getting into detail. And also, you can work on the interaction between the host plant and the vector by disrupting host selection process or making a plant less attractive, using repellents, trap plants, and other strategies. Plus, there are transmission barriers that can be exploited. And of course, this has to be adopted because the disease comes from the neighbor, and the neighbor may be right across the fence, but can be a little further. So area-wide management is a key component in this whole management strategies. Talking about disruption of transmission cycle, by target interactions. There are several things that can be done. So, and this is object of research right now by groups, different groups. So you can target the acquisition, trying to avoid Xylella ingestion. So this can be done using resistant material, for example, or feeding deterrents. And in plants, for example, there are some varieties that have impact on vector feeding. So, and they show less symptoms in the field. In Santa Catarina, for example, you can block retention by using vector receptor blockage mechanisms. I'll briefly show you something quickly. And also you can reduce inoculation by affecting vector landing on the crop, reducing through barriers, repellents, and other mechanisms. So you prevent landing and probing activities on the plants. One example is this use of kaolin treated citrus plants. So kaolin is a kind of a powder, a crop protected. It changed the color of the vegetation to a kind of a whitish color. So the leafhoppers are well attracted by the yellowish young shoots of citrus, but when you cover that with a white powder, it's not attractive anymore. And also, we know that some plants in the wild vegetation, they are very attractive to the sharpshooters. So we may plant them in the orchard edges close to native vegetation where you have reservoirs of vectors. So when they move from these woods to the orchard, they usually stop in these nice plants that are attracted, and this can be used as a trap plant. So trap plants and repellents can be used together in a push-pull strategy. Push, if the insects they come from neighboring areas, they instead of entering the orchard, because of this whitish color, they try to move away. They don't like it, so they stay on the trap plants that are more attractive. And here, insecticides can be sprayed and control the vectors before they enter in the vegetation. These two we have not tested thoroughly for CBC, but there are several studies using this for the psyllid that transmit citrus greening, and it's working. So the use of trap plants and kaolin. And we are quite certain that this will work well also in our case for the vectors of Xylella because they come from neighboring areas too. So these are some strategies that can be integrated to disrupt this vector-plant interaction. There are other ways that we can do to disrupt the bacterial vector interaction. We know that the bacterium has some ephemeral proteins, for example, that have affinity with carbohydrates in the vector foregut, some receptors in the vector. So this in red here is a kind of a membrane protein. The SHAD-A2 has affinity for these molecules. So if you somehow avoid the binding, you don't get, you disrupt transmission. In this case, a strategy was used by offering an antibody against this membrane protein. This was a study that did this kind of assay using artificial diets with these antibodies against this membrane protein by incubating the cultured cells with or without this antibody. And in the cases where you have the antibody, you have the reduction in acquisition rate and also in transmission rate, showing that you can block and disrupt, for example, affect the retention and subsequent transmission of the bacteria to the plant. Of course, this was done in the laboratory, in artificial diet, But these are things that could be exploited to affect, for example, retention. Okay. Anyway, this is examples on strategies that can be used based on the knowledge of the vectors. Francisco, how much time do I still have?
You can continue. 10 minutes, for example. Yes.
5 minutes.
I think
10
it's
minutes?
possible. Yeah.
Okay, so I'll just show a little bit of the situation we have in olives concerning vectors. So we are at a much earlier stage than we are in citrus, of course. We are in the stage of understanding what are the vectors and what are the predominant species and what vectors are transmitting better the bacterium. But we don't know well the host plant associations and several other ecological attributes. So we have a disease, we have a very severe disease affecting olives in the Mantiqueira mountain range region, which is a very nice, beautiful and touristic regions in São Paulo, in Minas Gerais state. There we have more than 100 species of vectors because we have these lots of wild vegetation. So sharpshooters, they love all these the woods. Many of them have hosts that are trees or shrubs, and you have a very high. This is rainforest, up to 1,200, maybe 1,400 meters. You still get rainforest, and then you start getting other types of vegetation. But you get a good mix of plants to sustain a high diversity of sharpshooter species. So more than 100 species were found, but we could test so far maybe 20 of them, and 14 were shown to transmit already to olives. Among them, some are predominant species based on faunistic analysis. So the similar work that has been done in citrus, we are doing in olives. And some species, they stand out as predominant based on faunistic indices such as dominance, frequency of collection, abundance, constancy of collections. So based on that, species like this Clastopteridae is quite common. Bucephalogonia xanthopsis, Erythrogonia, Maculonaria caviforms, Scopogonaria paula. Look at this one. This is predominant in our range of elevations here. Very common in the mountain range, as well as on Cometopidae. So these are very common species and constant and dominant species in the region. This is data of population density of sharpshooters based on yellow stick traps. So this is results of years of survey that indicates the mean number of insects per trap, per yellow stick trap. So you have here an average of Scopogonaria or Scopogonaria paula of 1 insect per trap, which it's a good population, reasonable numbers. And then these other guys here, they also have this higher occurrence compared to others. If you look on the plant, so this is a very important information because in many cases the host plant, the affected crop is the main source of inoculum. So those that are found on the canopy by using sweep nets, they are usually very important. And the same guys usually are also collected in the canopy of the olives. So Clastoptera, Maculonaria caviforms, Bucephalogonia, Scopogonaria. So they are among the most abundant on the canopy. And this one, especially the Clastoptera, which is a Clastopteridae, is developing in olives. So you see nymphs and you see adults. And populations, they peak late summer and fall, and they are found in several locations in the mountain range. So we believe that this is probably an important vector, although we still haven't tested yet as a vector. So if you put together all the ecological information that we have, if you rank them concerning population density in values that range from 0 to 1, we get like a grade for relevance. So Scopogonaria, Clastoptera, Macugonaria cavifrons, Erythrogonias, Cephalogonias, they all have considerable values. And we think that these guys probably are relevant in this epidemiology. Although these others might have an impact too. We still need more studies, especially on host plant associations. We know that they are some of our animals, but we don't know what other plants they are feeding, and if there are other plants that are hosts of Xylella in the ground vegetations. You see that we have a very diverse ground vegetation in this region, several herbs and grasses present in the rows of the orchard. And also you have trees and shrubs that are neighbors in these areas. So we should understand a little bit the role of these plants. And also we need to do epidemiological studies as done in citrus. In citrus, the epidemiology helped a lot to characterize the role of this underground vegetation, the role of woods, and also the role of infected olives in the spread of disease. So we don't have this for olives. We need that information. So to end the talk, these are my final remarks. So in general, in Brazil and other countries in Latin America, especially in the tropics, but also in subtropics, including Argentina, Paraguay, usually have high diversity of potential vectors of xylem feeders that belong either to Cercopoida or to Cicadelidae that can have the potential to transmit the bacteria. Several of them have been confirmed as vectors. And it's important to understand, based on to understand the relevance of these different species, based on their transmission competence, based on the natural infectivity and other ecological aspects, especially population density, mobility, host plant associations. That we need information on that. So this knowledge of vectors and interactions with Xylella, and also on disease ecology, is basic for managing the disease. It's based on this understanding that we could assemble the strategies that are now shown to be successful to restrain CDC in São Paulo state. And the last point is that vector interactions with other disease elements like the host plant and the bacterium can be explored as targets for disruption of these disease cycles. So we don't need to rely only on insecticides, but we have many other tools that can be integrated into the management strategy. With this, I finish this presentation and thank you for the audience. Thank you very much.
Okay, João, thanks for your talk. It's been clear, precise, and interesting. In fact, when you told me about the time you have to finish, I had said 10 minutes because I was enjoying the talk, and I will continue. Okay. So I think there are no questions, but I would like to put you a question. Possibly, I think you can give me, or you can give us an answer. When you have say this expression, a vector or the vector, that is quite interesting. You think that in the case of Italy in Apulia, it was the presence of the vector? I say by the explosion of the spread of the bacterium and the disaster that he has produced in the region?
Certainly. I think here you have a clear D vector. This Philemus spumarium is clearly D vector, and not only in Italy, but in several areas where it's being found abundant, and especially on the crop trees, because of the characteristics.
As you see, possibly not only by the vector, but also by all the situation, the plant, the weeds that they are in the crop.
Yeah, certainly, because it feeds on develops in the underground vegetation, like in the spring, and moves to you have all the climatic, the weather, the climate that have the summer, dry summers, that you have the olives, they move to the olives. So it's a very important, the behavior of this. It's not only the ability to transmit in the population, but it has the behavior, the mobility, and there's ability to take the bacteria from water hosts to olives. So I think it's a good example of insect that assembles the different ecological attributes to make the job of transmitting Xylella fastidiosa.
And you have talked about the importance of the weeds as inoculum for the bacterium. Do you think that in fact that's a very important role? They take a very important role in the epidemiology of all the
yeah, like in citrus, for example, this role, although it may exist, we think it's a minor role compared to the citrus trees for CVC, because the data doesn't show like a kind of pattern, temporal or spatial, that indicates, for example, the influence of weeds in the spread. Always you have an increased aggregated pattern along some initial foci of the infected plants, and other characteristics of the epidemiology too. And also, but in terms of experiments, inoculating weeds and so on, we have one good study that was done. These studies are not easy to be carried out. So because you have to inoculate plants, several weeds, and have them in the greenhouse, do the transmission assays, putting insects on them. So this is very complicated to be done. But the one study that did this was not able to transmit to citrus. It does not mean that it is not possible. It may occur, but in lower rates. And also it has to do with the cycle of these weeds in the tropical situation. They grow very fast. In Xylella, we know that it takes some time for Xylella to build up population in young leaves. The leaves has to mature, and then you have accumulation. But by that time, either the weed is senescent for annual species, or it's being mowed because they grow so fast. People have to mow the underground vegetation. So Xylella does not have the time to grow. But we may have some species of sharpshooters or maybe spittlebugs that like to feed right close to the soil level. For example, in California, the green sharpshooter likes to feed in the bottom, close to just above ground. So a sharpshooter that has this behavior, or a cercospora, might have a higher chance to recover that bacterium that usually goes to the bottom part of the plant. And we did find in alfalfa, in my sabbatical I did with Rodrigo Almeida there, we exploited the difference in population in alfalfa from the very tip to the bottom, and the higher population was right at the base of the plant, the part that was remained uncut after the field was mowed. So the bacteria has adapted for that. Yeah. There is a vector that likes to feed in that portion. So I think it's possible that if you do studies, even in Brazil, where you have this fast growth of plants, that we may find something. In olives, we don't know anything. It's possible that in olives, especially in this high elevations, a little colder weather, we might have effects of that.
Okay, so thanks. I think that the audience will be a little tired by now, so it is time to end the webinar. It seems that we still have a lot of work to do to get a good control of Xylella, but I think it has been a very fruitful session, although very dense. And events like this one, I think, can help to the dissemination and discussion with the very different groups, with all the information we have. And I think these are very important events. And finally, I especially want to thank the 3 speakers for their participations, and also thank all the audience in the webinar. And I would like to remind you that tomorrow we will be the second online session of the workshop, which will start at 2 p.m. Lisbon time. So good evening to all, and until tomorrow. Bye.
Bye.